Back, everybody. I'm Jessica Hasty, a research reviewer in the Center for Biologics Evaluation and Research. We'll now begin our afternoon by discussing the applications of omics on safety and efficacy for product development. As a reminder, please hold your questions for the Q&A panel at the end of the session, and for our virtual attendees, please add your questions to the Q&A box in Zoom. It's my pleasure to introduce our first speaker of the session, Dr. Björn Peters from La Jolla Institute for Immunology. Dr. Peters has worked on the development and validation of tools to analyze and predict which parts of the pathogen, allergen, or cancer cell are targeted by immune responses throughout his career. These tools aim to reduce the experimental effort required to identify molecular targets or epitopes of immune responses. These epitope identifying algorithms developed in his lab are used to characterize how immune cells from diseased cells differ in individuals from diseased individuals and healthy individuals, which helps us understand how the disease develops, how we identify potential targets for intervention to treat or prevent disease. Finally, the Peters Lab is involved in developing community standards for knowledge representation to promote interoperability and reuse of data. Today, Dr. Peters will present on the community resource his group developed for comparing Bordetella pertussis booster responses and the contest they host for predicting patients' vaccination outcomes. Dr. Peters, I'll hand it off to you. Thank you very much for joining us.
Thank you for the invitation. I had another meeting at the NIH, so it was really convenient to be here the day before in person rather than doing this by Zoom, which I always prefer. So these are my disclosures, and this is the team that is really part of it. It's a quite large team. It's the Kleinstein Lab at Yale, Grant Lab at UCSD, and the iLab at LJI, and the same as my lab as well. So I thought I'd start today's presentation with a bit of background on pertussis vaccination and the reason for the switch of the vaccine in 1996. And then now we've tried to analyze what's happening there with using omics technologies, and then how we use computational models to interpret the data that we're getting from these omics studies. So whooping cough is caused by Bordetella pertussis, which is normally a respiratory infection that has mild symptoms, but they can be quite long-lasting. Another name for whooping cough is the 100-day cough. And last year, there was actually in the fall, several people had that. It's actually kind of interesting that it's often unclear what you actually had. It's often not seriously diagnosed, but the real reason why it's an important disease is in infants. There's a significant fraction who have very severe disease, including death. And today, pertussis is identified by the CDC as the least controlled bacterial infection that we have vaccines for, and that's despite excellent vaccine coverage. So the vaccine originally introduced for Bordetella pertussis is essentially a whole bacteria inactivated vaccine, which was developed in the 40s and given, and you see this massive decrease in reported cases. And then in the 1980s, there were increasing concerns about side effects. That's actually a really interesting sociological study in itself. The initial concerns were similar to like the vaccines cause autism, about neurological effects in some people that never actually were confirmed, but that led to like public distrust of that vaccine. And at the same time, it did cause significant reactogenicity, so as in swelling at the injection site, fever, unwellness. So that led to the desire to switch the vaccine to a more modern vaccine approach of individually isolated antigen targets rather than having the whole bacteria ground up. And this is given together with diphtheria and tetanus in childhood. So now what then happened is 10 years later, cases started popping up. And people looked at all kinds of different reasons. I'm not going to go into the evidence here, but it was really tied clearly at this point to the introduction of the AP vaccine, which, while it was in the initial test, it was extremely effective at protecting people immediately. So 10 years later, apparently, your immunity wanes. Now, for an FDA crowd, that becomes clear that it's really a terrible thing. How do you do studies of your vaccines or approve vaccines if the difference or the downside of a vaccine only becomes apparent 10 years later? So we want to understand that. But how do you study that even? I mean, you can't get money for a study that says, okay, let's immunize some people with WP and AP, wait 10 years, and then I'll tell you what I find. So instead, this recruitment strategy, which was pioneered by Alex Sette at the La Jolla Institute, was actually quite simple, taking advantage of the fact that the switch from the AP to the WP vaccine in the US was completely in 1996. So if you take people who were born in 1995, they got the AP vaccine. If you have 1996 and later, they got WP before 1996 and later, they get the AP vaccine, and then people are due for booster vaccines. So we take blood before and after the booster vaccination. The booster vaccination you can think of as an antigen challenge of the immune system, asking how do you react to this? And then we asked for are there differences depending on what you were primed with as an infant decades earlier. And we found indeed, and again, this is Alex Sette who originally did this, that there was a difference in essentially the polarization of the immune response, where there's a more Th1 polarization in the people who were primed with WP, while there's a more Th2-like polarization in the ones who were primed with AP, and that was mimicking results in animal studies, including from Todd Merkel at the FDA, that showed similar findings. Obviously, in animal studies, you can do things that you can't do in humans, but that backed this up quite nicely. But then the question comes, okay, great, now we know what's 2 decades later, but what actually maintains this? How does the immune system keep on making a polarized response based on priming that happened 2 decades ago, despite continuous booster vaccinations with the same AP response? That's the vaccine. That's the only thing that's approved now. And despite environmental exposure, subclinical or clinical infections, etc. So how is this maintained? And we wanted to use essentially then in omics approach of asking what are these initial events that happen once you're getting the shot in your arm, what are the different arms of immune responses that happen that then push you towards maintaining and boosting an existing Th1 versus Th2 response. So this is the first cohort then we enrolled there, and essentially we focused here in this first one on the first 14 days of the study and performed, I should say that RNA-seq for transcriptomic, CyTOF for cell frequencies, this is all in the blood, antibody titers by serology, and in plasma proteomics. And so these are examples for what we found for the gene expression data, where, general problem with omics data is always, you have way too many variables. We have here close to 100,000 different or so readouts at every time point. So the first things we do, we cluster them together into groups of, in this case, genes that are co-expression, and then we're looking for kinetic patterns, the ones that are actually perturbed by the booster vaccination, and then we find these main groups here. And then we have, as another example here, the kinetics of the cell subsets, where we see different cell types being perturbed in their frequency found in PBMC post-booster vaccination. And when we integrate that, we find, for example, up here, that the classical monocytes, which have an influx at day 1 right after a booster vaccination, they correlate really, really well with one of the transcriptional clusters that also shows a peak at day 1. So ultimately, you have a very complex gene expression pattern that is completely explained by an influx of monocytes in your blood. That's actually going to be one of my points in omics studies. You have this high-dimensional data, and you shouldn't trust it blindly. You can explain tens of thousands of changes by something that you can measure with a complete blood test, right? You can monocyte frequencies. You can test that. You don't need omics for that. But omics, you will find the same things in omics, and it looks much more complicated, but it's actually not. So this is the advantage and disadvantage of omics. So the same thing for antibodies here. When we look at 3 different modalities, we see that plasma antibody secreting cells in the blood, the frequency of them, peaks at day 7, along with the expression of immunoglobulin genes in RNA-seq. And when you look at the serology of the long-lived IgG in your blood, you also see that it matches this, but it obviously has a very different kinetic. The gene expression and these antibody-secreting cells, they are transient, they go on and off, but they make long-lived antibodies that you can find in the blood and that then peaks actually at 14 days because these cells keep on making stuff. So this is the great thing about omics. You're getting different readouts that can actually, in this case, support each other if you really know what's going on. And a complex signature in one readout might be completely explained by a simple signature like, okay, this is antibody production, or this is an influx of monocytes in another. Now, we did all this to study AP versus WP primed individuals. And there, it became much more complicated. We did not see such beautiful signatures that differentiate all AP from all WP individuals. Here's an example of a difference we find, that on day 3, we find an increase in CCL3 production, but only in a subset of the AP primed individuals. Now, that was actually a first thought, like why would we expect that if there is such a defect in the vaccine response, that it has to be in every one? It's not that the introduction of the AP vaccine response, everybody got sick. We see an increase in cases, so it could be that there's just in some people, but obviously another explanation is that just by random chance, we had 4 people here that for whatever reason like to make CCL3. And so how do we distinguish this? Obviously, the obvious thing is you have to repeat. And one of the problems very often in omics studies is that all people do is cross-validation in some shape or form, but you're building your models in your mind of what's happening, and this is the kind of thing that is being reported over and over. You really need a completely independent cohort and measurements that are done after you're done with this. So the other thing is what does CCL3 production has to do with waning immunity? Obviously, we don't know. And in general, how do we now analyze this data? How do we ensure that the findings that we have generally line beyond just our study? And that's where computational models come in. They're an excellent fit to analyze omics data because actually everything you've seen today, you need computers to analyze omics data. It's not something where you can look through lists of tens of thousands of variables and interpret them. So if you have a computational model, that essentially it forces you to say, okay, if I have these inputs, this is going to be my output, and you can test that. And then you can compare different models and ask which ones are actually predictive. Examples of models are gene signatures. People say if you express a lot of these genes at the time of vaccination, like inflammatory signatures, there have been a lot of papers about this, that's bad for your predicted vaccine response. There are mechanistic models, like viral kinetics over T-cell responses, etc., or here MHC antigen processing. And there's black box models, such as the neural network, where we just feed all the data in and do your favorite AI algorithm and get some kind of answer back. It doesn't matter what you do, though. If you have a model that really predicts something well, then you understand your system better. You can query that model, essentially, why are you predicting this, even if it's a black box model, and understand better what's happening and develop new and testable hypotheses much faster in your model, then you can run more trials. So our goal with the computational model of immunity pertussis boost project was to develop a research to enable these steps of model development based on omics data. And this is our workflow. So we essentially take a study cohort, we take blood samples pre and post boosters, we do the multi-omics profiling like I showed you. Then we provide participants with this training data from the previous years, like complete data sets. And then we tell you, here are our new participants, and we just tell you their baseline state. This is their immune cell frequencies and everything just before they got the vaccine. Can you now tell me what is going to be your influx of monocytes on day 1? Is it going to be higher or lower than others? What is your CCL3 production on day 3, which we have as a potential marker of AP? What is your IgG titer against pertussis, which is the traditional readout that people want to see for vaccines? And then we repeat this. We do the same study every year with a new cohort and over time increase our data as we know it and ask people, can you now predict for the new year what's going to come out? And the first year, we just did a dry run, just internal people. Second year, we invited some contestants to participate. And in the third year, it's an open challenge, and everybody can participate. So in our readout is essentially we take, say the monocytes on day 1, we rank them in our data sets, and then we take the model predictions. We say, okay, based on your model prediction, these are the highest, these are the lowest. We do a Spearman correlation. That's our performance measure. So the dry run results were published recently, and this was the setup. So the first thing that we noticed was that this is actually harder than we thought. We said, okay, we're just going to do the same experiment every year. Ends up you can't, because companies start making kits. You suddenly have to switch your instrument breaks. You have to go to a different instrument. So specifically working on cutting-edge multi-omic studies, this happens all the time. And it's a real problem. I mean, a trivial example is like there's a wealth of data out on microarray studies that nobody can reproduce anymore because nobody runs microarrays anymore. It's all RNA-seq now. And that data is completely different. So even if you're saying, I'm studying gene expression on X, the way you study is really important. So in our case, obviously, we tried as much as possible to keep it constant, but essentially, we can only connect the things that we could do the same. Then we took all the literature models that are out there. There hadn't actually been any for pertussis, but there's a wealth of people who are saying we can predict vaccination in general, and things like this inflammatory signature has been reported in multiple cases, so we just implemented those and said, what do those models say? And then we had our own team 2 and 3 that tried different prediction algorithms, and these were the outcomes from our dry run. So the published models from the literature, that again, like they were developed for different vaccines, so they didn't claim to predict pertussis booster vaccination, but it was still fascinating that none of them worked. Not a single one, one worked. And that was actually a control model that used calendar age of the subjects. So after all this data, the one thing that predicted a poor vaccine response is if you're older. Please, somebody laugh. That is not a good new finding, right? But that's a really important control, because if you can make big omics data sets where all you actually have in there is an age signature, and with 10,000 parameters, etc., you will essentially predict age. And you could have just taken age. Okay. Our custom approaches work better, but for example, we didn't realize that age thing. We didn't have age in our own parameters, or not in all of them. But we did find the other thing that is also kind of trivial, in the end, right? Once you have the data, everything becomes trivial, is that your pre-vaccination baseline data is highly predictive. If you have really high antibody titers against something, pre-booster vaccination, guess what? Your antibody titers are still going to be more likely to be higher than someone else. So, and then this one algorithm performed better, and we're worried a bit about time. So we learned from this, right? Then we did our second challenge, where we now have 96 complete longitudinal cohorts, and we have 22 new. We have the same prediction tasks. Actually, we had more at the start. We learned we should make it simpler. These are the 3 that we stuck with. Then we separated. Actually, that's another key thing that we learned is what do you actually use as your metric of what is a good antibody response. Some studies say it's the increase of antibodies that you get by the booster. Others say, I don't care about the increase. I want the level at the end. Which one you pick is really depends on what you're interested in, right? If you have, and what ends up being, if you have a really, really high antibody titer, you're not going to boost it anymore. Probably because also the antigen is just immediately removed based on your existing antibodies removing it, right? So if you were looking at full changes, having a low antibody titer makes it more likely that you're going to get a good response in terms of full change. So we invited 50 people in addition to the previous contestants that were publishing in this area. We had also found out that the whole work in harmonizing the data that we have is really crucial. There's a lot of pre-processing work to get your data into the format that the bioinformaticians like it, namely a big matrix. They don't want to know what the different measures are so much. They just want to have a number next to a variable name, and that's it. So we have these outcomes here. We got 28 submissions from 18 teams. overall, it really worked because we obviously shared with them, this is what worked last time, try to improve on it. So everything became better, much more significant submissions. And the winner was a team from Minnesota, which is also awesome. So completely, people had nothing to do with us, had never worked on pertussis vaccination before, outperformed the others. And one of the key things that they did differently from most others is that they went back and redid all the data and harmonization that we did using a different approach. They specifically imputed all missing data with quite nice effects, and they did different batch correction. Batch correction is the other really terrible thing with omics data. If you run things over multiple years, again, on different instruments and stuff, or just the operator changes, all kinds of things changes that you have no idea of. They are going to impact your data, and you have to correct for it somehow. So they used actually a similar computational approach, and overall really showed good results throughout. So again, I'm worried about time, so I'm going to skip the summary. Actually, one of the key problems that we had is like we only had these 22 new subjects that ends up to be a little bit too low. We have variability in the data. To have better rankings, we want to have more results. So in this third public challenge, which was just announced like a week ago or so, we now have a lot more new people. We have, now, where is it? Something like 50 new subjects. Oh, yeah, 54 for the prediction. Now there's 117 complete training sets with 10 time points each, all this different omics data. So this is an excellent way to learn. So to summarize, the lessons learned here is what makes omics great is that we're getting different insights from different modalities of the immune system that ideally can explain each other and give us more confidence in the findings that we have. And in the same pattern that might be complex in one might be simple in another modality. Then computational models, okay, the high number of variables that we have make it important to carefully analyze them. It is really, really easy to find something in your data set that is never going to repeat in anyone else's. Computational models are an easy way, I think the only way, how to interpret this data compared to others. The need to repeat studies is important to test model performance. This is hard with omics data, as I said. Data harmonization is an underappreciated problem, as in people tend to focus on the algorithms later. But how you deal with your data before it comes into the algorithm is more important in my mind. And then stringent control models. It is not sufficient to say that your model predicts better than random at whatever significance. You need to say, I'm better than using the last good model and minimal things like age and the baseline status of the individuals that you have. And with that, I'm slightly over. Thank you.
Thank you, Dr. Peters. Next, I would like to introduce Dr. Robert Teresky from the Mason Cancer Center and Department of Medicinal Chemistry at the University of Minnesota. Please go ahead and start sharing your slides while I do a brief introduction. Dr. Teresky received his PhD in Nutrition and Food Science from MIT, and before his current position, he served as the group leader of the Biomarkers Unit at Nestle Research Center. He also served as the Division Director of Chemistry at the FDA National Center for Toxicology Research and was a principal investigator at the Wadsworth Center for New York State Health Department. His research investigates the biochemical toxicology mechanisms of potential cancer-causing agents in the environment, tobacco, food, cosmetics, dyes, and traditional herbal medicines. Dr. Teresky uses mass spectrometry approaches to identify and measure these chemicals, their metabolites, protein and DNA abduction products in laboratory animals, cell culture, and human biospecimens. Today, he will present on his novel technologies and mass spec-based approaches that his lab has developed to screen for DNA abducts from a wide variety of genotoxins, which provides a comprehensive data on genome-damaging chemicals that contribute to cancer. Dr. Teresky, I'll hand it off to you, and thank you for joining us today.
Yeah. Well, thank you very much for the introduction and for the kind invitation. I'd like to cite my colleagues, Scott Walmsley and Pete Vialta at the University of Minnesota for their excellent collaborative research with me. I have no declarations to make or conflicts of interest. So the outline of my talk is, well, what are DNA addicts and why do we measure them? Then I'll talk about some of the common methods of detecting DNA addicts and focus particularly on mass spectrometry. And then I'd like to give some examples of some of the DNA addict screening applications that we have employed in animal models and human tissues, including some work on aromatic amines, bladder and liver carcinogens, heterocyclic amines, which are multi-site carcinogens in rodents, potential human carcinogens, aristologic acid, which is an FDA-regulated compound, and some lipid peroxidation products due to oxidative stress, and just finish up with some of the challenges in the future research. So historically, traditional epidemiology has been working in a black box. It's been mostly an observational science. However, in the last decade or 15 years or so, particularly because of the advancements in mass spectrometry tools, the research has become much more mechanistic rather than observational. So for example, we can measure chemicals in tobacco smoke or carcinogens in grilled meat at parts per billion levels or lower. I understand some aspects of exposure, but what we really want to know is about the internal dose, which we can measure through urine, blood, accumulation of compounds in hair or fingernails, for example. But we really want to know is what's the biologically effective dose. That means the critical metabolite of most carcinogens, which do require metabolism, and their chemical damage to DNA and proteins. Ultimately, what our goal is to understand what DNA addicts form that induce mutations and can be linked to specific mutational spectra or signatures in the genome, strengthening the associations of exposures and chemicals involved in cancer etiology. So this cartoon here, again, recapitulates that most carcinogens require metabolism, and broadly speaking, they can be broken down to two different pathways. Fortunately, most of the compounds undergo detoxification, but in some cases, a portion of the dose is unfortunately going to be activated to a reactive intermediate that can latch on to DNA to form a DNA addict. Fortunately, we have many DNA repair systems in our bodies that can remove these DNA lesions before cell division occurs. However, unfortunately, in some cases, some addicts are refractory or do not efficiently undergo repair, and thus during cell division, they can trick the polymerases to create a mutation in a codon. And if that's in a cancer driver gene, after other multiple biological processes, you can result in having a cancerous cell. So DNA addicts are internal dosimeters of genotoxic exposure. They're used in interspecies extrapolation of toxicity data and mechanisms of bioactivation of a carcinogen. Identifying DNA addicts and assessing their mutagenic potential are among the first steps that are used in assessing the role of a chemical in cancer risk and etiology. Some DNA addicts can be linked to mutational signatures. I need to emphasize, however, if we examine the tissue for a DNA addict and we don't find it, doesn't mean that that individual was not exposed to that carcinogen at some point in time, but either through repair processes or because of cell division, that lesion is no longer detectable by mass spectrometry techniques. So that's an important point. So DNA addicts often represent recent exposures, whereas the mutations that they induce accumulate over time during exposures, particularly those that occur chronically. So there are many methods to examine DNA damage. I'm highlighting perhaps the three most importantly used techniques that have been used over the last 3 or 4 decades, including P32 post-labeling. So the DNA is digested down to the nucleotide level, where a polynucleotide kinase will transfer a radioactive P32 isotope, very high radioactivity, to the 5-hydroxymethyl group of this modified DNA base. And in this case, two-dimensional thin layer chromatography is used, followed by autoradiography. And you get kind of like a diagonal zone. This is actually DNA damage from the lung of a smoker. This technique shows that there's DNA damage, but it's very rare that you actually can identify what the specific chemical is that has modified the DNA. That's the weakness of this methodology. There immunoassays have been developed with DNA that's been modified with the carcinogen, and monoclonal and polyclonals have been developed in either animal models or cell systems now. This technology is useful because you can actually identify within specific tissues or cell types by immunohistochemistry potential lesions. The weakness of this assay is, one, there's not many antibodies that are available. Two, even monoclonals, the specificity is questionable and could be, the antibody could be cross-reacting with a number of different components, including some cell constituents. It works wonderfully in animal models. Its validity in humans really needs to be cross-validated by specific mass spectrometry methods to ensure there's no false positives. Mass spectrometry techniques, including gas chromatography and liquid chromatography, are perhaps the most powerful and specific methods for identifying addicts. So carcinogens or the metabolites are electrophilic, and they can react with many different positions on the nucleobases and resulting in a complex plethora of potential DNA addicts. So again, the use of high-resolution mass spectrometry is the state of the art to look at DNA addictomics and trying to screen for multiple types of carcinogens in the diet and the environments. So the first scanning technology I'm going to talk about is ion trap mass spectrometry. So you have a liquid HPLC system which chromatographically separates different addicts. They are collected in the ion trap. In this case, we're doing targeted work and we are breaking this molecule apart. And that gives us, we can scan out in the ion trap and detect the loss of the deoxyribose, which is 116, and usually get the A-glycone, represented as H2 here. Or we can collect that fragment ion and put it back, keep it in the trap and fragment it a third time to get characteristic A-glycones, product ion spectra, which is more or less a fingerprint of the addict and is essential for identification. More recently, Orbitrap high-resolution mass spectrometry has been established. I'm not going to get into the configurations of this and explain it. I don't have the time, but basically, this instrument allows you to detect things at parts per million levels instead of at nominal mass or 0.1 Dalton, making the instrument and the technique much, much more selective and specific. So again, we can measure DNA addicts by targeted MS to the third, where we take the precursor ion, we break it apart, take the iClone and break that apart to the MS to the third, to give us a product ion spectrum that's characteristic of the addict. However, that's targeted. We also wish to do untargeted methods, and we have two techniques that we use. One is called data-dependent acquisition. In this case, what the instrument does, it will collect, say, the top 3, top 5, or top 10 peaks as it scans, and the instrument's instructed to fragment these, and subsequently, it looks for those precursor ions that have lost the deoxyribose, and it'll do MS to the third. This is an excellent technique. However, it's biased towards the most abundant ions. So if you have addicts that are present at low levels but biologically relevant, that scanning technology may not work. We've developed another technique which we've adapted from proteomics and metabolomics using swath type techniques, where instead of taking 1, 2, or 10 ions, we'll take everything within a packet of, say, 30 MZ window and fragment everything in there. And then we will detect those at the MS to the second stage. Then post-acquisition, we extract putative precursor ions and fragment ions which have lost deoxyribose at very high mass accuracy, the neutral loss of 116.0473. So for an example of some of the untargeted work we've done, this is 4-aminobiphenyl. It's currently its most prevalent source of exposure is through tobacco smoke, but also some environmental pollution. It's a liver carcinogen and a bladder carcinogen in rodents, and it's thought to be definitely a bladder carcinogen in humans, and perhaps also liver carcinogen. So we examined its metabolism and DNA addict formation in human hepatocytes. We have an untreated specimen here, and we have the treated specimen here. And you can see at the DDA, the conjugal neutral loss, MS to the third, we're not seeing any addicts detected in untreated cells, but we have 3 peaks detected in those cells treated with 4-ABP, and we were able to acquire the mass spectra. This major peak is the deoxyguanosine adduct called DGC8-ABP. I had a standard of that. I expected to see that. However, the instrument also detected these 2 other addicts, which I don't have reference standards for. The third product ion spectra give beautiful product ions that help in interpreting and elucidating the structure of these unknown addicts. Let me switch over to aristolochic acid, which is an FDA-regulated compound. This chemical is present in traditional herbal medicines. This is actually an outdated slide. The number of cancer instances in these different countries is much greater now, but this became a public health issue in the early 1990s when a group of women in Belgium who were otherwise healthy were taking a slimming regimen, which unfortunately contained Chinese herbs containing this renal carcinogen. Many of those women developed renal failure and or renal cancer. So it was noted that the pathologies that was observed in these women was very similar to individuals living in the Balkans, where the wheat fields are commingled with contaminating Aristolochia herbs. Thus, the bread that was produced was contaminated with this carcinogen, and there's extremely high incidences of renal disease and renal cancer in this cohort. In China and in Taiwan in particular, Aristolochia has been used for many years, centuries, as part of a regimen in traditional medicine, and there is very high instances of renal cancer in these cohorts. So historically, this adduct has been detected by P32 post-labeling methods. We have established, and I believe we're still the sole lab in the world that actually screens for this adduct by specific mass spectrometry methods. So how does this compound get bioactivated? Well, again, it needs metabolism. In this case, it undergoes nitroreduction to form the N-hydroxylactam, which undergoes phase 2 conjugations to form this N-sulfate, which is a labile bond that generates, after hydrolysis, nitrinium carbenium ion. This reactive intermediate binds to adenosine at the N6 position of adenine to form this adduct, which is quite stable, actually. And under DNA replication, the polymerases get tricked and they put an adenine instead of the normal thymine codon across the adduct. And during the second round of replication, what you have is an AT to TA transversion. So Professor Grohlman, who I've had the honor to collaborate with at Stony Brook University, what he did is he took the IARC database and saw that this AT to TA transversion is quite minor. However, in the cohorts of Taiwan and Balkans, they exceed 50%. Again, this is an uncommon transition. He also looked at the P53 gene, mutations in the P53 gene, which is a tumor suppressor gene, and he superimposed the codons which were mutated in the Balkans and in Taiwan. As you can see, they're almost superimposable. These cohorts reside over several thousand miles away. However, this is very strong chemical and molecular evidence that the same causative agent is causing the cancers in both cohorts. We've done a number of targeted mass spectrometry studies in Taiwan, high frequency of the addicts, and then in these different cohorts, lower but appreciable levels. And again, the chromatograms above with the internal standards show that this individual did not have the adduct. This is our lower limit of detection. And this individual had higher levels of the adduct, and this shows the mass spectral data corroborating the identity of this lesion without any ambiguity. Since this work was done, Alexandrov, who developed these single base signature mutations, and this signature 22 is highly characteristic of aristolochic acid, and many of the subjects in this cohort showed this signature, reinforcing the notion that this is a critical compound that these groups are exposed to and develop cancer. We followed up some work with Ghislain Scello at IARC. Looked at 4 different cohorts, the Czech, UK, Russian group, and as well as Romania. We did this blind coded. All the subjects who had renal cell carcinoma were positive for this adduct in Romania, but none of the subjects in these cohorts which did not manifest the disease were positive for the adduct. In addition, the proportion of AT to TA transversions increased as a concentration of the adduct in these tumors. Again, supporting the notion that this chemicals contributing to renal cell carcinoma. So this is targeted work. Let me talk about this WIDESIM. This is the second data independent technology that we use. And my colleague Scott Walmsley developed an algorithm because we had to do this all manually, extracted ions manually. And he developed an automated feature where we would identify the precursor ion, its N+1 isotope, as well as the aglycone and its M+1 isotope. And with various spectral and chromatographic features, he would automatically pull these out for data for adduct identification and data analysis. So let me jump to an animal study. We did a study with 13 carcinogens which may contribute to renal cancer. And one of those compounds that we tested was aristolochic acid. We expected to see this adduct. And again, this is our internal standard marker. But we have this other major peak which suggests it's an isomer. I've never seen this adduct before in any other study. We also have a deoxyguanosine adduct of aristolochic acid. But again, there are two other major peaks which appear to be isomers we've never seen before. Well, as it turns out, in this study, we actually used a commercial extract of Aristolochia herbs. Before, we've only used pure chemical standard. What we've seen is that in addition to aristolochic acid, there's a minor peak in here, an isomer with the methoxy group is 2 positions over. And as it turns out, the mass spectra of aristolochic acid in this new putative isomer clearly show they both have the aristolactam product ions at 292, 93, indicating indeed this is an aristolochic acid isomer. We proved it with subsequent studies that it is AA3. We also identified some of these deoxyguanosine adducts. Their mutagenic potential is uncertain. We don't know. So this WIDESIM technology, we've adapted to human tissues. And as an example here, this is, again, our untargeted work on aristolochic acid. We do an MS to thirds to corroborate the structure. In this case, we're looking at FIP. This portion of the this adduct is derived from a cooked meat carcinogen, a heterocyclic amine, which we think contributes to human prostate cancer. This is extremely challenging work. You can see we have to zoom in to look at the signals of aristolochic acid. This is a mirror plot of the synthetic standard. This is really literally pulling a needle out of a haystack. And we need to go to the MS to the third stage to corroborate adduct identity. This shows the challenges we have in looking for adducts. We've applied this technology to look at prostate cancer, low and high Gleason score patients. And these are, again, some of the adducts that we've identified. We've actually identified formaldehyde, indicative of oxidative stress present in this lipid peroxide fraction. We have the FIP adduct, which is in the aromatic fraction, and a number of lipid peroxidation products. And again, the high and low Gleason score subjects clearly separate out. Challenge we have is most of these putative adducts, we don't know what their structures or identities are. This is the challenge in advancing this technology. Retrospectively, we can take the data that we have, it runs already over, and we can go back and ask, well, do we have adduct X, Y, or Z? If we know what their masses are, we can just plug in those numbers and we can extract out their precursor ions and their product ions. And you can see there are a number of lipid peroxidation adducts that we've seen that are elevated in prostate cancer patients with high Gleason scores, which is an aggressive pathology, suggesting oxidative stress and inflammations occurring. So in conclusion, I know I went through this very quickly. There are many putative adducts exist in the genome. Challenges, most of them are not known. Untargeted adductomics that we've used identified several novel adducts. AA3, is it potentially existing in human cohorts and contributing to renal cancer? These screening techniques can be used with humans, but with caveats. The signal of response of adducts varies greatly, which is going to affect the sensitivity and level of adduct detections. No adductomics method is going to capture all adducts. Some of the challenges for the future is future biopsy samples are not available. Oral cells, white blood cells, exfoliated cells are, however. The method is a slow throughput. It's labor intensive. You can semi-automate the DNA isolation. Improvements in adduct detection and removal of interferences is required, and there are various techniques that can be done to do this. High resolution mass spectrometry, the scanning rate's kind of slow, which can limit the coverage. However, new instruments are coming on the market and they are more rapid scanning, which will expand the coverage. Data analysis and identifying unknown addicts remains a challenge. We need the help and collaboration of bioinformaticists to develop algorithms to help us analyze the data and also to help deconvoluting overlapping peaks with chimeric spectra and mass spectral interpretation. This is a developing technology in human biomonitoring. I'd like to thank my colleagues and also B.H. Jun. I don't know if he's here today listening. He's left our group 2 years ago and wonderful scientist, and he's now, I'm sure, doing a great job with the FDA. The funding, our colleagues, the Grohlman Group, the group in Thailand, the patients in particular, our funding from NIH, and I'd like to thank the Masonic Chair in Cancer Causation at the University of Minnesota for their support of my research. Okay, thank you very much.
Thank you very much, Dr. Tereski. Our next speaker is Dr. Sandra Fuentes at the Center of Biologics Evaluation and Review at the FDA. She obtained her PhD in pathobiology from Pennsylvania State University, where she investigated the function of RSV phosphoprotein and small phospholipid protein. She completed her postdoctoral fellowship also at the FDA, where she contributed to the development of a luciferase reporter gene based on microneutralization assays for RSV and investigated the antibody repositories of RSV-infected and vaccinated individuals to identify protective epitopes. She is currently a microbiologist here at the FDA, where she contributes to the development of reference materials for the evaluation of high-throughput sequencing platforms aimed at the detection of advantageous vaccines. Today, she's going to tell us about her high-throughput sequencing potential for endogenous virus detection on multiple Illumina sequencing platforms. Dr. Fuentes, please take it away.
Thank you, Jessica, and good afternoon, everyone. So my talk today is about cell standards for adventitious virus detection in cell substrates and cell-based products used in high-throughput sequencing technologies. And first, a disclaimer, this presentation is an informal communication and represents my own best judgment, and all comments I make here are my own and do not bind or obligate the FDA. High-throughput sequencing, or next-generation sequencing, has proven useful for the detection of known and novel viruses in environmental, clinical, and research biological samples. Adventitious viruses are a major safety concern in biologics because they can be unintentionally introduced during the manufacturing process as exogenous viruses due to their presence in the biological raw materials. They can be introduced during the handling, or they can be present in the equipment and facilities. They can also be present as pre-existing endogenous viruses in the cells. During the manufacturing process of biologics, HTS is recognized as an advanced technology that can be used for adventitious virus testing in cell banks, virus seeds, and the bulk harvest. And it can be used in these instances to replace or supplement in vitro assays, especially when there's viral interference with the cell that you use for your study. To replace in vivo assays, which can reduce or eliminate the use of animals. The same when you're looking at rodent antibody production tests, using HTS can reduce or eliminate the use of animals. And it can also be used to replace PCR assays, which can reduce the testing time since you'll be doing one assay instead of multiple PCR assays. But before you can use STAs as a method for virus detection and product safety, the method needs to be validated. And for that, reference standards should be used for evaluating the entire HTS workflow, from the sample preparation all the way through the bioinformatics. So our lab, the Arifakant lab here at CBER, has developed various reference materials and resources that can be used for HTS method validation. And the topic of my talk are the cell-based standards, but we also have a reference virus database, which you can find in this website, and it's database of all viral sequences, excluding bacteriophages, but including endogenous retroviruses and retroviral elements. And it has a reduced cellular contact, and so this database facilitates the analysis of HTS data for adventitious virus detection. We also have a panel of reference viruses that can be used for evaluating the sensitivity and breadth of detection of your HTS pipeline. And so we have two versions of this panel. The first version included 5 viruses. It includes Epstein EBV, fetal leukemia virus, human respiratory syncytial virus, human reovirus type 1, and porcine circovirus. And all of these viruses come from different families that have different particle sizes, from very small, like PCV1, to large, like RSV. They have envelopes. Some of them don't have envelopes, and they have different types of genomes. So it includes double-stranded DNA, linear or circular, single-stranded RNA, linear or dimeric, or double-stranded RNA. They also vary in their size, the genome size, so from very small, like PCV1, to large, like EBV. And finally, they have differences in their levels of physical-chemical resistance from low to high. So this panel of 5 viruses has since been discontinued, but it's been replaced with a panel of 7 viruses. This the panel of 5 viruses can still be found through VEI at the catalog number that I have in this slide, but it was replaced with a panel that includes these 5, but also a human coronavirus, OC43 strain, and the Munich virus of mice. This panel, as I said, can be used for evaluating the sensitivity and breadth of detection of your high-throughput sequencing method. And they should be used as a panel to evaluate the qualification, verification, or validation steps. But the topic of my talk is the cell-based standards. These are virus-infected cells that can be used to evaluate the sensitivity of detection of HGS for virus contamination in cell banks or cell-based products. And these were developed by infecting human A549 cells with a simian foamy-virus, and they vary in the level of viral RNA and virus production. The main focus of my talk is the 55C cell line, which is a cell line that has a latent infection of SFB. So it has viral DNA in its as a provirus in the genome, but it only has very basal levels of our RNA expression. And you cannot recover the virus by either infection of the cell supernatant into a permissive cell line, or by co-culture of this 55C cell line with a permissive cell line. In addition to the latent clone, we also have 2 clones that have varying levels of viral RNA production and virus production, so the 51C and 1C, and I'll talk more about them later in my talk. But as I said, the focus of my talk is this 55C latent virus, and why would we be interested in latent virus? So it's still a safety concern in biologics, because the latent virus infection results in the persistence of the virus genome with little or no transcription for virus production, but these viruses can be activated or reactivated during the manufacturing process. So latent viral genomes are can be integrated into the host cell DNA, or exist as extrachromosomal epitopes, circular or linear DNA in the cell. And they include endogenous retroviruses that naturally occur as stable integrants in the host genome. The majority of these genomes are defective, so they're missing large chunks of the genome, but some of them are full length and can be expressed as infectious virus. It also includes viruses that maintain their latency as epitomes, including herpes virus, human papillomavirus, and adenovirus. And these can occasionally also be found partially or completely integrated into the host genome. So our goal was to use simian foamy-virus as a model for latent virus infection of a human cell line. So simian foamy-virus is a non-pathogenic retrovirus that naturally occurs in all non-human primate species. It has a broad tissue tropism and cell tropism, including human cell lines. And when you infect cells with it, it can result in a latent infection with no virus production, chronic infection, where you're going to get continuous virus production or viral RNA expression, but the cells continue to grow fine, or it will result in a lytic infection where the virus kills the cells. And the outcome of the infection depends on the virus strain and the cell line used. For our study, we used human A549 cells that were infected with a naturally occurring strain of SFB that we isolated in the lab. Its designation is SFB-K3T. When you infect A549 cells with this virus, it results in a latent infection. So to generate our latent clone, the 55C cell line, we used these K3T infected A549 cells and cloned them by limited dilution. And this resulted in many clones, and we evaluated them for virus integration and persistence of the virus in the host DNA, viral RNA expression, and virus production using RT activity assays and looking for infectious titer. So what we noticed is that when we clone the cells, we get different phenotypes. We've classified it into three. The first one is a chronic infection where you have the virus genome integrated into the cell. You also have virus production, and in the case of these chronic clones, you're going to have also virus in the supernatant, so you can isolate the virus from the supernatant in most of these clones. You're going to have RT activity, and you're going to have detectable viral RNA expression. For the persistent virus clones, you also have virus genome integrated. We are going to have a low level of viral RNA expression, and you're going to see viral RNA expression inside the cells, but you're not going to recover it from the supernatant of the infected cells. Now, for the latent clones, you also have the virus genome integrated, but only basal genome expression, and you can't recover the virus either by infection or by co-culture into a permissive silent. So it's a truly latent clone. And this is the clone that we wanted to use to evaluate latent virus, detection of latent viruses by HTS. So we further characterize this 55C cell line by determining the number of proviral copies that were integrated into the cell. We also determined the integration sites by TLA, and we looked at viral gene expression and RT activity by doing a PERT assay, and we tried to determine whether we could isolate the virus, as I mentioned before, either by co-culture and infection. So by all measures, this cell line was latent. We cannot detect RT activity or detect the virus. And we know that it has 2 copies of the K3T genome per cell. So the objective of this study was to evaluate HTS for the detection of the latent viruses using this model of retrovirus infection in the human cell line. And we evaluated different HTS platforms for the sensitive detection of the latent virus, tried to identify the number of reads that gave us a valid positive result, and in general, assess the potential for using latently infected cells to determine the limit of detection of latent viruses, detection by high-throughput sequencing in cell substrate and cell-based biologics. So the design of the experiment was very simple. We just spiked 55C cells at 10 to the 5 to 10 to the 1 cells per 1 million cells of the uninfected A549 cells, extracted the DNA and used that to prepare the library and sequence by HiSeq. In general, we obtained between 600 and 800 million reads from each sample, which we then used for quality checks and discarded the reads that were that failed the quality filter and the size filter. And we also removed the reads that were mapping to the human genome to aid in the downstream analysis. So the remaining reads were used for targeted analysis, which is where we map the reads to our reference K3T genome, or by non-targeted or agnostic analysis by doing a blast N of the viral RBDV. So this is a summary of the results for the targeted analysis of the sample, and as I said before, we have between 600 and 800 million reads per sample. We were able to detect virus in the 10 to the 5, 10 to the 4, and 10 to the 3 spikes. And the coverage of the genome varied between 88% in the 10 to the 5 spike to only 2% in the 10 to the 3 spike. This last was important because if we repeat the study with the 10 to the 3 spike, we only detected in 1 out of 3 experiments. So these 2 reads were a perfect match and would have required a follow-up experiment, but it's not reproducible. And in our hands, the limit of detection for this cell line in our workflow was 10 to the 4 infected cells in the background of 1 million infected cells. And this is just to show you the type of coverage that we got with the different spikes. For the 10 to the 5, we have most of the genome, most of the K3T genome is covered by the reads. 10 to the 4, you can still get good coverage and a confident, valid positive result. For the 10 to the 3, we only have 2 reads, and like I said, the results are not reproducible, possibly due to sampling issues at this level. So after the targeted analysis, after we determined the limit of detection by targeted analysis, we wanted to see what was the limit of detection by the non-targeted or agnostic analysis. So we start with the same pool of breeds that we started with the non-targeted analysis. We collabed the identical reads and used those reads to do a blast N against our reference virus database. Then the hits that we get from our reference virus, against our reference virus database are divided between what we expected to find and everything else which was unexpected. And the only thing we expected to find is whatever is also in our negative control. All the other hits were followed up by determining which were the reads that aligned to these unexpected hits, and then doing a blast analysis against NT. These reads were then classified as viral or non-viral. And I'm just going to show you a summary result for this experiment. So all of the spikes had non-viral reads. These reads are mostly just host sequences. It varied from human to other species, but they were all host sequences. We also had not determined reads, which were reads that had low complexity sequences and couldn't find similarity in the BLASTN analysis. And finally, our viral reads, which were found in the 10 to the 5, 10 to the 4, and 10 to the 3 spikes. We also had 2 viral reads in the 10 to the 2 spike, but follow-up analysis showed that they aligned with the same specificity to different species of virus, plants or fishes, so they were really not viral reads at all. And all our viral reads in the 10 to the 5 and 10 through the 10 to the 3 spike were also identical to our spike virus. So we were able to detect our spike virus down to the 10 to the 3 spike. But again, these results were not reproducible at the 10 to the 3 level, only at the 10 to the 4 spike. So by both the agnostic and targeted analysis, we identified our limit of detection for our workflow to be the 10 to the 4 infected cells. So finally, we wanted to evaluate the effect of the depth of sequencing in the detection of adventitious, in the detection of the virus by HGS. And so for that, we used the 10 to the 4 spike because we had already determined that it was positive by HiSeq reproducibly. It was also positive doing the ddPCR analysis. And so we resequenced using NovaSeq SB, which gives you about 1 billion reads. The NextSeq MEAD platform, which gives you between 300 and 400 million reads, so it's a little bit lower than what we get with the HiSeq. And the MiSeq V2 technology, which gives you between 30 and 40 million reads. And surprisingly, we were able to detect the virus using all platforms, although, as expected, the higher the number of reads, the better the coverage you have for your virus. And so we suspect that if we repeat this experiment with the MiSeq, we might see it in some cases and not in others. But these 2 reads were identical to our virus and would have required some follow-up analysis. And again, this is just to show you a representation of the coverage that we got from the different platforms. You get better coverage with the NovaSeq than the NextSeq and the MiSeq. So to summarize, HGS was evaluated for detection of latent viruses in spiky studies using A549 cells that were latently infected with simian foamy retrovirus. And if we use HiSeq, the limit of detection of the latent virus was 1 times 10 to the 4 infected cells in the background of 1 million uninfected cells by both the targeted and non-targeted approach. We did find 2 reads with perfect alignment to the virus reference that were sufficient for a positive result in the 10 to the 3 spike, but these results were not reproducible, possibly due to sampling. We also looked at the effect of sequencing depth and on latent viral detection by MiSeq, NextSeq, and NovaSeq, and all 3 platforms were able to detect the latent virus in the 10 to the 4 spike. Although, as expected, lower sequencing depth resulted in less viral reads detected. And we believe that this cell line could be used, can be a useful reference reagent for in-process control for the whole host high-throughput sequencing workflow to determine the limit of detection of latent viruses in cell substrates and cell-based biologics. And the last thing that I want to discuss is that we also have cell standards for transcriptomic HGS. As I mentioned in the beginning, we have two clones, 55C and 1C, that vary in the level of viral RNA expression. The 51C has low level of viral RNA expression, and you can only recover virus by co-culture of the cells with permissive cells, so there's no virus in the supernatant and no variants. And the 1C clone has higher viral RNA expression, and you can recover virus from both the supernatant and the cell. So we're currently doing an internal spiking study similar to the one that I just showed you to determine the limit of viral detection by transcriptomic HGS. And finally, we also have a collaborative studies through NIMBLE with the goal of optimizing high-throughput sequencing virus detection in a complex biological matrix, so using an unprocessed bulk harvest. And for this study, we will be using not just the CBER cell standards, but also the WHO reference panels that I showed you in the beginning. And our goal is to try to optimize the HGS protocol to get a more sensitive detection in a very complex background. So with that, I will end my talk. And I want to thank Arifa Khan, who's the driver of these projects and the PI of the lab. Paige Sheen and Trent Bosma formed the lab, are in charge of the RBDB maintenance and annotation. And Belita, Terry, and Hannah were instrumental for making the cell clones and their characterization. And also the CBER core facility and CD Genomics for doing the high-throughput sequencing analysis. So thank you.
Thank you very much, Dr. Fuentes. As our final speaker of the session, it is my pleasure to introduce Dr. Jeff Bouquet of New York Langone Health. Please go ahead and start sharing your slides while I do a brief introduction. Dr. Bouquet obtained his PhD from Rockefeller University, and then he did a postdoctoral studies with Gerald Fink at the Whitehead Institute. He has since then gone on to found or co-found several biotechnology companies and is the founding director of the Institute for Systems Genetics at NYU Langone Health, which is known for its foundational work on the mechanistic and genomic aspects of retrotransportation. As a pioneer of synthetic genome construction, he has synthesized the first artificial yeast chromosome de novo and leads an international consortium that built the highly engineered genome of the first synthetic eukaryote yeast 2.0, which synthesized 17 synthetic chromosomes to a common design and invented genome scrambling. He will present his work on very deep omic profiling of samples from decedents who have undergone xenotransplantation of kidneys and hearts, as well as developing clinical-grade PCR and sequencing tests for the detection of porcine exogenous and endogenous viruses. Dr. Bouquet, I'll hand it off to you, and thank you very much for joining us.
Well, thank you very much for having me. Can you hear me? Can you see the slides?
Yes, we can.
Great. Well, before I begin, let me just say that this work represents a new direction for our laboratory, and it is a major collaboration with Brendan Keating and Robert Montgomery in our Department of Surgery. And you can see my disclosures here. And in the picture, you can see the first pig heart, genetically engineered pig heart that was transplanted into a human decedent in our surgery department here. And of course, this is a potentially game-changing technology that meets a huge unmet need. So my talk today will cover that topic, as well as explaining a little bit more about what Xeno, as I'll call it for short, is and how it works. It sort of burst on the scene even though it's been tried for over 100 years unsuccessfully. In 2021, it burst on the scene when Bob Montgomery did the first transplant into a decedent here at NYU Langone. And this decedent model, which, not to put too fine a point on it, is transplanting a pig organ into a brain dead individual. is kind of a game-changing way to molecularly evaluate the risks and benefits of this trans-species transplantation method. So I'll tell you a little bit about our first evaluations on the very short-term effects of the xeno kidney transplantation on the PBMCs and tissue samples from the individual. I'll talk about how we've been doing some assay development for xenoviruses and what our results have been. And I'll end with a study of a 61-day decedent multi-omics study and some of the prospects for diagnostics and prognostics that we think are going to be required once this technology becomes approved for clinical trials, which we think may be not so far in the future at all. So about the unmet need, only 1 in 4 patients currently on the waiting list will receive an organ. And it's very clear that there are many other people who could greatly benefit from an organ, but aren't even on the waiting list because it's such a hopeless situation for people awaiting a transplant. The increase of obesity in our society, as well as the plateauing of organs that are available for transplant, result in over 100,000 on today's U.S. wait list. And I want to emphasize that there's a particularly big unmet need in underrepresented minority populations and economically disadvantaged populations, which of course suffer disproportionately from obesity and diabetes. And so xenotransplants could massively reduce healthcare costs by reducing dependency of some of these individuals on dialysis. So pigs provide significant economic and clinical advantages for human xenotransplantations. Their organs are similar to ours, but the animals require only 5 to 10 months to reach a size where they can be used. They have many offspring per litter, and they're relatively easy to engineer their genomes. I won't talk about that aspect at all, but that's something that we're actually quite interested in. But there have been at least 6 gene editing companies that are in this space and operating in advanced stages here in the US and in China primarily. So I'll mention both a single gene knockout model, which is the alpha-gal knockout, as well as the so-called 10 gene pig. So there's more than one 10 gene pig, and the exact set of genes is not the same for every source of these animals, but many of the genes are the same. So the first 3 knockouts, GGTA, CMAH, and B4GAL and T2, all are genes that encode glycans that are on the surface of pigs and other mammals' organs, but are not present in primate cells. And so this is what the GGTA1 encodes the alpha-gal. It makes the alpha-gal, and this has been pinpointed as the major cause of so-called hyperacute rejection. So I'll have more to say about that later. So every engineered pig that's out there has this knockout. Then in addition to that, the growth hormone receptor is sometimes knocked out because, when you have a 5 to 10 month old pig, the organs may keep growing and get too big. So that's why that's there. And then there's a whole series of transgenes that have been knocked in. Primarily, these are epithelial cell certain surface proteins that interact with complement and interact with the blood system and the immune system in various ways. And I'm not going to talk about all of that, but this is basically what's been engineered. And then in 2021, Dr. Montgomery and soon after others, rocked the world by the concept of using a brain dead individual as the recipient, as a way of testing xenotransplantation kind of radical therapy. And the huge advantage here is the deep accessibility and profiling of tissue and blood that just isn't possible in conventional living human studies. The potential use of experimental medications, and kind of a fail-safe environment for developing protocols for xenotransplantation. Some of the challenges, besides the fact that finding decedents who are eligible to do this and getting buy-in from their families at a very difficult time of an unexpected brain death. Obviously, that's a major challenge. The cause of death may be a confounder, and it's very expensive. It's done in an intensive care unit setting, and obviously there's some regulatory complexity. So, so xenotransplantation involves the use of genetically engineered animals. Here I'm showing some purple DNA, which came from human, has been inserted into the genome of the pig. And so now that pig organ that we're going to transplant into eventually a living patient is going to be expressing human proteins on certain surfaces of that organ. There's a second wave, we think, coming, although it's been slow, which is so-called exogenesis, in which it's been shown that this can be done in rodents, that you can take an iPSC in principle from a person. So this would be the purple cell here is the human cell. You can inject it into the blastocyst of a pig, and these cells can engraft into the embryo. And if this pig has been engineered so that they can't, for example, develop a kidney, then those human cells would take over that niche in the animal, and you would actually grow an organ that is 100% human, or essentially 100% human. And not only that, but it's a perfect match to the patient himself or herself. And so that is really going to be a game changer if that can be made to work. But I used to think that was closer than it is, and now I think it's farther away than it is, because this engraftment is quite inefficient. Okay, so what is a decedent? It is a brain-dead organ donor who is medically unable to provide an organ due to cancer or infectious disease status. The decedent family opts for whole body donation for a period typically lasting 72 hours to as long as 61 days. And as I said, just about 3 years ago exactly, the first one of these was performed. And one of the other reasons I'm very excited about this is because it solves what I like to call the three-body problem, which is that what we're really interested in here with xenotransplantation is taking an organ from a pig and transplanting it into a human. But until this model arose, really all the tests were done with a third body, which is non-human primates. And it turns out that the immune systems and the reactivities and so on of non-human primates are similar, but definitely different from the human response. And so here we're obviously not going to have a third party. So this is a accurate, as of a couple of months ago, summary of all the xenotransplants that have been performed. They've all been performed in the US and China. And as you can see, there are 6 transplants that were done at our institution, but increasingly we see some that have been done in hospitals in China as well, University of Maryland, University of Alabama, and UPenn. This is moving very fast, and there have now been, as you can see, 5 live transplants here, and some of these patients have gone, I guess the record holder here is 65 days. And that's progress. Obviously, we'd hope to see things go longer than this, and there are many reasons why these particular individuals didn't last as long as we might have hoped, and we're learning a lot from the decedent studies about maybe making it more successful as we move along. So I'm going to tell you just a little bit here about the very first transplant that was done. This was less than 72 hours in length. It was a pig to human kidney xenotransplantation, and this is the simple simple alpha-gal knockout pig. So just one knockout, and this, as I mentioned before, this has been shown to block the major hyperacute rejection response, which results from the fact that 2% of our IgG is raised against this glycan, alpha-gal, which is very common part of bacterial cell walls. So we're all exposed to it, and we all have this massive load of IgG against this glycan. And primates are the only animals that don't have alpha-gal on their surface. So it's not like, this is kind of a unique situation of going from an animal, a mammal, another mammal into humans. So one of the bolder elements of this plan was to go with this single knockout rather than the more advanced one. But in fact, now all 4 of our kidney xenotransplants were performed using the it's called the GalSafe pig from Revivicor United Therapeutics. And these, interestingly, they were actually developed for people with the red meat allergy. This is, you may know that this has been spreading because of the spread of the Lone Star tick. This somehow sensitizes some people to this red antigen, which is obviously very common in red meat. And so these pigs were actually developed for people with this red meat allergy and FDA approved as food in 2022. Another important fact about these organs is that they're not a simple kidney. They're what's called a thymokidney. And the way that you make a thymokidney, as I understand it, is you take a small fragment of the thymus and you embed it under the kidney capsule about 5 months before the animals are to be used. And work from Megan Sykes and David Sachs over many, many years has shown that this will help to tolerize cellular immune responses. And so perhaps this is, we don't have any proof of this, but we think that this actually is one of the things that is making this quite successful. And so we don't see any evidence for hyperacute rejection, despite the fact that the other 2 glycans that are not found in humans were found in these animals. Okay, so what did we find? We didn't find signs of hyperacute rejection. We found evidence of endothelial cell and immune response activation, early signs of antibody-mediated rejection. This is just a 72-hour study. And very interestingly, within 12 hours post-xenotransplantation, we found evidence of human immune cell infiltration in both of these xenografts. So here you see the tiny red spots here in the single cell analysis from the tissue. The human cells have invaded the kidney, and similarly here we see in the PBMCs we see a mix of different cell types. So, whoops. And in the PBMCs, we see a biphasic response of immune responses at 12 and 48 to 53 hours. So these global expression signatures also showed xenotransplantation-associated kidney tissue damage in the xenografts, but then also evidence of a potential healing response in the form of some proliferative cells in the xenografts, indicating early activation of some kind of tissue repair program. Another major part of our work in this has been to develop good assays for porcine viruses, xenoviruses. And this is based on, during the pandemic, we actually developed a highly scalable qRT-PCR test for COVID-19. We actually worked with a company here, a local company here, Opentrons, to set up the pandemic response lab, which actually ran 11 million of these tests. And based on that technology, we developed assays for 8 porcine viruses of concern. You can see them listed here, as well as some of the control that we developed, and that the assay performance is quite good. The results of this have been insanely boring, in that essentially we never, in all of these studies, we haven't detected any of these viruses, except, as expected, we find the PERV, porcine endogenous retrovirus, readily in the porcine samples, which of course we expected. But very importantly, we saw essentially no evidence for the transfer of the porcine endogenous retrovirus to human. In the few cases where we could detect it in the PBMC, we also detected the mitochondrial DNA from the porcine tissue, and we interpret that as cell debris contamination of the PBMCs. We always see those two things go in hand, the mitochondria and the porcine endogenous retrovirus. And of course, this method doesn't detect unknown viruses, and we are working on sequencing methods to detect those. And then last but not least, we did the 61-day decedent study. This is one-time thing where we performed extensive multi-omics in the form of various RNA sequencing, including spatial RNA sequencing. You can see that we regularly sampled both the tissue and the PBMCs. And we did very extensive genomics, as well as proteomics, metabolomics, and of course, clinical measurements on this one individual. And I should emphasize that this individual,
1 minute.
the study was terminated not because of death, but because we had agreed in advance that that was as far as it would go. All systems were still functioning very well at the end of the 61 days. And here you can see that this individual, like a lot of transplant patients, developed a number of infections due to the immunosuppressive regimens used. So, of course, we have very deep data, clinical data on standard blood tests and urine tests. And in addition to that, we did a lot of omics, and I'm just giving you a taste here. This is the Seer proteograph, which uses a magnetic bead that is incubated with the serum. And this protein is, protein corona is then used to identify the peptides that are present in the serum. And remarkably, we can detect 8,200 different proteins from the serum. The vast majority are human, 43,000 peptides, but we also see 11,000 pig peptides, and we see quite a variety of pathogen-derived peptide signatures corresponding to some of the same pathogens that were detected in the individual. And at day 33, we saw a big change in one of the protein modules that emerged from this study. And this was also reflected in changes we saw in RNA-seq, single-cell RNA-seq, and immune cell typing. This day 33 was clearly a peak of a rejection episode. He had all these signatures of immune cell rejection. And one of the other things you can see here, these bars are 33, day 33, is that a lot of the complement proteins were tanking at that time. And there's a whole coordinated series of molecular changes consistent with a rejection episode, which was successfully managed clinically. We also see major fluctuations in different immune cell types over this 61-day period, flickering up and down, sometimes somewhat coordinately, but often with very different changes in different compartments of the immune cells. And last but not least, we are closely studying cell-free DNA in the samples from these individuals, which we can sequence on Oxford Nanopore. And on day 33, there was a huge peak in the proportion of the DNA that came from the pig organ. And we think that the peaks here that are above the background strongly suggest to us that these could be prognostic predictors of forthcoming rejection episode, which could be an extremely valuable diagnostic test. So in conclusion, genetically engineered pig organs, even if relatively lightly engineered, can be transplanted without major risk of short-term rejection. The decedent model provides a wealth of information on the human response. Multiomics, and especially proteomics, discovers many signals that could be valuable for monitoring rejection and infection. The exception to not detecting viruses in our studies is the PERV, which is expected. We did see no evidence of infection of human cells despite high levels of immune suppression. And the methods we deployed did not detect unknown viruses, but we are developing sequencing methods to detect those. And last but not least, a huge thank you to the decedents and their families that allowed us to perform these studies. My major collaborators, Bob Montgomery, Brendan Keating, Bo Shah, and Adam Griesemer and their research groups, as well as members of my lab and the total team, which consists of over 100 people. Thank you very much.
Thank you very much. Please join me in welcoming Dr. Bouquet and all of our speakers this session for very excellent presentations. Unfortunately, we are out of time for question and answers, but I encourage those of you who are on the Zoom to put your questions in the Q&A box, and we'll have our panelists answer those virtually. And if you're in the room, please stick around for the break. Two of our speakers are present today, and I'm sure they'd be happy to answer your questions during the break. So thank you for everyone for attending this session. We'll take a brief break before beginning the final session today at 3 p.m. A reminder, there are coffee and small snacks available at the kiosk outside. Thank you.
I'm Carlo Mercado, an animal biotechnology reviewer in FDA's Center for Veterinary Medicine, as well as co-leader of the FDA Omics One Health subgroup. We'll now begin our final session today on leveraging omics for surveillance, emerging threats, and One Health. I'd like to introduce our session's first speaker, Dr. Scott Jackson from the National Institute of Standards and Technology. As a reminder, please hold your questions for the Q&A panel at the end of the session. And for our virtual attendees, please add your questions to the Q&A box in Zoom. Scott Jackson joined NIST in May of 2014 after 11 years as a principal investigator at FDA CIFSAN. At CIFSAN, his research focused on characterizing the global genomic diversity of enteric pathogens with applications for food safety, bioforensics, and public health. At NIST, Scott is the founder and leader of the Complex Microbial Systems Group. In his current role, Scott is leading efforts to improve NGS-based measurements of complex microbial communities, aka microbiomes, with applications in biosurveillance and infectious disease diagnostics. Scott performed his graduate studies in the biochemistry and biophysics departments at the University of Maryland and Johns Hopkins University, respectively, where he focused on the evolution of mobile genetic elements. Scott performed his undergraduate studies in chemistry and geology at the University of South Carolina. Scott will present on the NIST partnership with stakeholders from industry, academia, and other government agencies to develop standards for pathogen detection as it applies to infectious disease diagnostics, environmental biosurveillance, biothreat surveillance, and biomanufacturing quality control. Dr. Jackson, I'll hand it off to you, and thank you for joining us.
Carla, thank you for that very nice introduction. Thanks for the organizers for inviting me. As you might have heard Carla say, I worked at FDA for 11 years. It was a very proud time in my life. I started here as a postdoc in 2003, and when I was putting my slides together for this talk, I came across some old presentations from my FDA work, and I just want to spend 1 minute taking a stroll down memory lane of some of the work that we did here at FDA. Out until the late aughts. So we used high-density microarrays, the Affymetrix platform. We developed custom microarrays that had millions of probes that were complementary to genes and enteric pathogens, the salmonellas, the E. coli, et cetera, that cause foodborne outbreaks. From this, we could do gene detection and gene-level discrimination. This is not a gene expression. It is a gene detection. So we're hybridizing genomic DNA here, and those scatter plots you see show gene differences between strains. So we're strain-level discrimination. And if you notice that microarray on the bottom left, half of it says tiling. So we use probes that walk along the genome, and that allowed nucleotide-level discrimination. And what we could see are SNPs that were present in the genome. So high-level resolution that allowed us to produce these phylogenetic trees and do outbreak investigations in epidemiology and all of that practical stuff that the FDA does. Working with NCTR, Weta Tong's very talented bioinformatics group, we wrapped all of this microarray information into their ArrayTrack platform that was like an easy button for data analysis. So, you know, it integrated all of the probe sequences and annotations, and you could take a raw data from a microarray, plug it into ArrayTrack, and get these very nice analyses done at the push of a button. So a couple papers we published there. I had the opportunity to work with dozens of very talented, wonderful scientists while I was here, but I just want to highlight one person in particular, my partner in crime, Isha Patel, who I worked with pretty much every day, attached to the hip, as you see here on this. So as Carlos said, after 11 years here at the FDA, I moved to NIST. I was given the opportunity to build a new program there in microbiology. It took me about a year to kind of get my head wrapped around what NIST is. A lot of people aren't familiar with NIST, so I want to take a minute to introduce you to NIST. NIST is the National Institute of Standards and Technology. We're a non-regulatory federal government agency in the Department of Commerce. So key point there, NIST is not a public health agency. We are in the Department of Commerce. So we're the oldest technical institute in the federal government, dating back to 1901. We have two large campuses, one in Gaithersburg, Maryland, just 10 miles from here, and another in Boulder, Colorado. I bet if you look closely at those two pictures, you can tell which of these campuses is Boulder, Colorado. About 5,000 scientists and engineers strong, about $1 billion annual budget. And the NIST mission, and this goes back to our role in the Department of Commerce, our mission is to promote U.S. innovation and industrial competitiveness by advancing measurement science standards and technology. You know, the earliest practical application of measurement science was commerce, that if we want to trade goods, we have to agree on some measurement platforms. Here's a fun fact that'll make you feel good about your tax dollars. Every dollar invested in NIST has returned $9 to the U.S. economy, according to independent studies, and that was published in a science article last year. So there's a lot of negativity about government these days, but here's something that will make you feel good. Okay, we talk a lot about the bioeconomy. So yes, we work with biopharma companies, we work with biotech companies developing clinical applications just like you all do. We just sort of do it under a slightly different umbrella, slightly different direction, but the work is oftentimes indistinguishable. So to highlight the role that NIST plays, I want to point back to 2013, when this monumental thing happened that the FDA authorized the next-gen sequencing instrument as a clinical diagnostic tool. And it was such a big transformation that the director of the NIH at the time, Francis Collins, and the director of the FDA at the time, Margaret Hamburg, wrote this piece in the New England Journal of Medicine highlighting this monumental approval. And in it, they stated that contingent on the FDA's approval was the necessity for NIST to develop reference materials that are the best possible quality to benchmark the analytical performance of this device. And this initiated the NIST Genome in a Bottle program. Many of you may be familiar with this. It's the Human Genome Sequencing Quality Assessment Program, and it has some of the most well-characterized human genomes available. Biotech companies that are developing targeted NGS assays for clinical diagnostics are using the NIST human genome standards to validate those assays. So here's a cartoon that really highlights how we interact with FDA and industry. Remember, the industries that we work with, that we're developing standards for, are regulated by the FDA. It's important for all of us to come together to discuss what benchmarking materials are needed, how do we validate, what is a proper validation, and if NIST can play a role in developing some of the standards for that, then that's a win-win-win for us, FDA and industry. So the NIST microbial program is it's about 10 years old now. And we have two major directions: microbiome, clinical applications of the microbiome, being complex microbial communities, and the other side being biosurveillance, a close kiss detection. So the way I see these two things, microbiome versus pathogen detection, in one case, you're characterizing the entire haystack, and in the other case, you're looking for a needle in a haystack. But what brings these two together are NGS-based metagenomics. And in case you're not familiar, in NGS-based metagenomics, you have a complex sample. Maybe it's fecal material, maybe it's soil, maybe it's cerebral spinal fluid that's infected with something. The point is you don't know what's in it, and there could be many microbes in it. You just do a DNA extraction, and you put that DNA on your sequencing instrument, and you get some reads off of it. And from this, you go directly from sample to sequence data in a culture-independent way, because every organism that we know of on Earth is nucleic acid-based, whether it be DNA or RNA. You will see the signatures of those organisms in the sequence data. So you take the raw sequence data, you then cross-reference it against some genome database. Maybe it's NCBI, but there are many. There are custom databases, and what you're doing is you're trying to assign a read to a taxa to say that some organism is present. You only see what's in your database. I'm going to date myself, but back in the day when we had phone books, and if you wanted to find someone's phone number, you looked their name up, and then you found their phone number. This is like a reverse lookup, and if that person isn't in the phone book, you're not going to find them. Once you've done this, you can assign taxa and relative abundance, and I'm sure you've seen these kinds of plots, chronographs, et cetera. But it's a quantitative and a qualitative approach to identifying complex microbial communities in given sample types. You've heard about this already from Jeff and from Sandra, but NGS-based metagenomics, it's untargeted, and untargeted, that you take that in the context of PCR, that is a very targeted, you have a PCR approach and it looks at a specific target, and if that target's there, then it'll amplify and you see it. And that's opposed to this untargeted approach where you sort of see everything because you're putting all of the nucleic acid on the sequencer. This has been referred to as an agnostic diagnostic, and it's being adopted across various fields and disciplines, including infectious disease diagnostics, epidemiology, environmental biosurveillance, biothreat detection, biomanufacturing quality control, as you just heard Sandra talking about very nicely, with adventitious agent detection. So it's moving its way into high-stakes applications that fall under regulatory authority. So this is the metagenomics workflow. As you can see, it's a little more complicated than the 101 slide I showed you a few slides ago, where it's just sample DNA sequence. There are many steps that start with sample collection and storage, all the way through nucleic acid extraction, sequencing, and bioinformatic analysis. I like to refer to metagenomics as the 21st century microscope because it allows us to see things that we couldn't see before, just as the microscope did 300 years ago. But there's bias in every step, from how you collect and store your sample to how you analyze and interpret your data and everything in between, and there are a lot of things in between. This is a fishbone diagram, also known as the Ishikawa diagram, and it's just a way of visualizing and representing methodological variables that can and do impact your results. So, sample collection and storage, there are plethora of proprietary storage buffers and sample collection kits, and store at -80, store in ethanol, store it frozen, lyophilized, et cetera. Sequencing instruments, library preps, nucleic acid extraction, there are many, many, many methodological variables here. So a couple of years ago, we partnered with some industry groups, including Janssen's Human Microbiome Institute, the big pharma company, and a couple others, and we launched this big international interlab study. Some of you here at FDA were part of this and are represented in this co-authors list here. But what we did is we made some human fecal samples and we sent them out to about 50 labs around the world, told them to all measure them and send us the results back. It was a systematic analysis of the impact of the methodological variability. So all of these things, we wanted to assess the impact in a rigorous way. And I could talk about this for days, but what I want to show you is really this slide. And when the participating labs in this interlab study were running the samples, yes, it's important for them to process the samples and analyze and generate the data and send to us, but just as importantly was the metadata. We wanted to know every little granular, nuanced detail about how they did their analysis, how they ran their methods, where did they store the samples, what DNA extraction kit did they use, what vortex instrument, what vortex machine did they use, what power setting was it on, how many seconds did they vortex. We wanted to know every little nuanced, granular detail of their methodological variables. So we had a standardized metadata reporting sheet. It had about 100 questions on it. These graphs that you see on the right that I know you can't read, I can't read from here, but every one of these little gray boxes is one of those methodological variables, one of those questions, and what we did is sort of like an ANOVA on steroids, where we looked at the impact of that methodological variability on the result. Out of the about 100 questions that we asked, each one of these in statistics speak is a factor. We found about 60 that had a significant impact on the results. So whenever you see a red blip in this sea of data, that is a level within a factor that had a statistically significant impact on the result at the 99.9% confidence interval. I'm sorry, I think I said earlier that 60 of these had statistically significant impacts. That's incorrect, 39 did. I know you can't read this, so what I'm going to do is zoom in on that bottom left corner that I have highlighted, and this is these were the questions that were asked of the interlab participants. So let's look at the bottom left. What was the sequencing machine manufacturer and model? So out of the hundreds of data sets that we got back, you can see that, and yes, we didn't label the y-axis because a lot of these were long-worded responses, but majority of people used the same sequencing instrument. The 100 there that you see probably, as you could guess, is the Illumina MiSeq, but other platforms. And again, the point here is that methodological variables at every step of the way may or may not have a significant impact on the result. This was published recently. Yep, published recently, as I showed you there. I just want to point out some conclusions to this interlab study that we branded as the Mosaic Challenge. So there are a multitude of methodological variables and metagenomic next-gen sequencing measurements that can and do affect the results. So remember this little workflow that I showed you? Let's do a thought experiment real quick, and there are 6 steps shown here. Let's say for each of these 6 steps, there are 5 methodological variables. So for example, DNA extraction is the second step there. Let's say you have 5 kits, and you want to assess those 5 kits. And you move on to library prep, and you have 5 kits, and you want to assess those. So for every one of these 6 steps, you have 5 variables. If you do the quick math on that, that's 5 to the 6th power, which is greater than 15,000. 15,000 possible ways to measure a microbiome. This is conservative, and I guarantee you every one of those will give you a different result. So this begs questions about reproducibility and validity and validation and regulatory oversight. These methods are inaccurate. They might be precise, reproducible, if you use the same method over and over without varying any of your methodological variables. You can get good reproducibility, that's precision, but they are not accurate. We're not getting the right answer. If your result tells you it's 2% E. coli, probably not 2% E. coli. We don't have ground truth for a lot of these. So these interlab studies are important tools for understanding the impact of these methodological variables. I want to rant for a minute and just say that a lot of people describe metagenomics as unbiased. I just did a quick PubMed search here for unbiased metagenomics, found dozens of articles. Here are 4 of them that I'm highlighting. Metagenomic NGS is not unbiased. It is the opposite of whatever unbiased would be. Every step introduces enormous bias. I can appreciate the spirit of what these authors are saying, that it's an untargeted method, so therefore you're not biasing yourself with any specific PCR targets. But I would like to subscribe to the community that let's agree to use untargeted instead of unbiased. So one of the things that NIST is most well known for is developing reference materials. If you visit our headquarters building in Gaithersburg, in the lobby, you'll see a display cabinet that this picture was taken from, and you can see that we have standard reference cigarettes up at the top left corner. In the middle there, we have standard reference peanut butter. We've got Portland cement, and we've got industrial sludge and domestic sludge. Some of these might sound silly, but every one of them was developed for a very important industrial application. It takes a small team of PhDs, on average about 5 years, to develop a reference material at NIST, so this is not something we commit to lightly. We do a lot of basically market research to understand who needs the standard, how are they going to use the standard, how is it going to advance the industry. And going back to our roles with FDA, back in 2016, the FDA Center for Devices released this guidance document on their current views on using next-gen sequencing as a tool for infectious disease diagnostic. And in this guidance, they said that NIST is currently developing microbial reference materials that will be valuable tools for use in evaluating new diagnostic devices. So this launched, this was, going back 8 years ago. This launched our development of what is now known as RM8376. It's a NIST reference material, quantitative microbial DNA. Sometimes we refer to it as the mixed pathogen DNA reference material. It is not mixed. These are individual components of 20 organisms that you can see there on the left. But the point of them is that you can make mixtures, complex mixtures that are quantitative and qualitative. You know taxonomically exactly what's in there, and you know quantitatively exactly what's in there. So that you can develop ground truth mixtures to benchmark your NGS-based infectious disease diagnostics. So this was developed in collaboration and with funding from FDA Center for Devices. This is Jason Crawl at the bottom right, standing in front of our 4-degree fridge. This material is stored at 4 degrees of about 700 units of this material that we produced. The box that you see in the bottom middle there is what you will receive if you purchase this material from NIST. And as again, it's got 20 components in it, 20 tubes of DNA, plus a human component. The human component there is included to build mixtures that mimic real clinical samples that are often contaminated with human DNA. So we don't just make reference materials, but we like to do use cases to sort of demonstrate their utility and show the community, the industry, how to use them. So in its use for infectious disease diagnostics, what this graph is showing are different sample types that are often collected for infectious disease diagnostics, and we're plotting the complexity of the sample on the y-axis versus the ease of collection on the x-axis. And as you can see up in the top right there, stool is very complex with hundreds of taxa, and it's easy to collect because everybody poops. Down at the bottom left there, in the middle, you see CSF, cerebral spinal fluid. So it is it should be sterile, and it's of low complexity. So what we did is, how am I doing on time, Carla? You wrap it up? Okay. So we used our RM8376 in real stool, in real CSF. We made complex mixtures, dilution series, and we showed the analytical response of detecting these pathogen genomic signatures in complex real matrices, that is human stool and cerebral spinal fluid. Interesting here, spiking these organisms in CSF, which is a relatively simple matrix, versus stool, which is a complex matrix. It had the same response function, that is y equals mx plus b, the slope and the y-intercept were the same. So the matrix complexity doesn't appear to affect the sample, the response functions, although the limits of detection, as you can see highlighted in the squares there, the limits of detection were a couple orders of magnitude greater for the CSF, the simple material, versus the stool, as intuitively you might expect. Okay, if you like these kinds of conversations about method validation and understanding bias, I encourage you to go to IMSA.org. It's a consortia that we formed that meets quarterly, and we just have conversations with invited lecturers about measurement assurance around NGS. My group at NIST gets a lot of funding from other federal agencies to develop standards. I mentioned that we receive funding from the FDA Center for Devices, shown here, but we also receive funding from CDC, EPA, Homeland Security, DARPA, and other Department of Defense agencies that support our standards development in partnership with these agencies' needs. Here's the team of very talented people that I have the honor of working with every day, and that's it.
Good. Thank you, Dr. Jackson. And our next speaker is Dr. Helena Solo-Gabriel of the University of Miami. Helena, please go ahead and start sharing your slides now as I briefly introduce you. Helena Solo-Gabriel is a professor of environmental engineering at the University of Miami, where she teaches courses in water quality, environmental analysis, environmental engineering, microbiology, and water and wastewater treatment. Her area of research expertise is in problems that relate human health to the environment. Through this line of research, she evaluates the transport of microbes in the environment and assesses infectious disease risk. Most recently, her team utilizes wastewater-based measurements of the virus that causes COVID-19 to track the transmission of disease within the University of Miami, within Miami-Dade County Public Schools, within the university hospitals, and within Miami-Dade County. Most recently, her team has expanded workflows to evaluate the transmission of additional illnesses through measurements at regional wastewater treatment plants. Dr. Solo-Gabriel will present the applications of wastewater for tracking COVID-19 and its use in evaluating other non-respiratory illnesses such as Mpox and Candida auris. Her presentation will include workflows for expanding wastewater-based epidemiology beyond SARS-CoV-2. Dr. Solo-Gabriel, I'll hand it off to you, and thank you for joining us today.
Thank you so much for the introduction. Do you see my first slide?
Yes.
Okay, thank you so much. Thank you. My name is Helena Solo-Gabriel, as mentioned. I'm a professor at the University of Miami, and I will be describing our project. It's a joint project with 3 PIs from both the University of Miami and Weill Cornell Medicine. Stefan Schur is another PI from Miami, and Chris Mason is the PI from Weill Cornell Medicine. What I'd like to describe is the work that we've done looking at wastewater omics technologies to evaluate illnesses in the community. Our funding comes from the National Institutes of Health, RADx Radical Program, also from RADx Up, which is underserved populations, and also from 4Catalyzer. Our aims of our projects are to combine human clinical data with wastewater data within a data integration platform so that we can use that information quickly to characterize outbreaks as they are occurring. Our hope is that this information would be useful for policymakers as they mitigate and prevent future pandemics. I want to begin with acknowledgements. This was a huge team effort. University of Miami was the only university in Florida, South Florida, when the pandemic first hit that stayed open. We are a private university. All public universities were closed. And we attribute this to the fact that our president, Julio Frank, also an expert in public health, put together a very aggressive program for monitoring illness on campus, and brought all units of the university together. And we were able to also bring in Weill Cornell Medicine to help us keep the student population safe throughout the pandemic. And as a result, there were very many units of the university that were involved in this program, including the University of Miami Sylvester Comprehensive Cancer Center, the University of Miami Hospital, the University of Miami Center for AIDS Research, the Miller School of Medicine, my home school, the College of Engineering, Weill Cornell Medicine, and the Metasub Consortium. Our sample analysis program, or our workflow, is illustrated here. We begin by collecting samples of wastewater from the sewer system. Those samples are then brought into the laboratory, and then we split them 3 ways. One split went to the Center for AIDS Research, where Dr. Mark Sharkey developed a new method called Volcano Second Generation, qPCR, which has a very rapid turnaround time where we were able to collect samples in the morning and have them analyzed by the afternoon. This technology circumvents the need for making cDNA. It uses a novel polymerase that can use RNA as a template. In addition to the work at the Center for AIDS Research, also we had rapid turnaround through the Oncogenomics Shared Resource, which processed the samples using the more traditional RT-qPCR, and we use that to check the V2G qPCR. Also at the Oncogenomics Shared Resource, we had samples deep sequenced for variant analysis. And then through Weill Cornell Medicine, our samples were sent for shotgun sequencing and metatranscriptomic analysis. All the bioinformatics that are shown here on the sequencing data were done by Weill Cornell Medicine. Our sample collection program was guided by where we could get data that was of human significance. So on the right, we have Miami-Dade County, and we have data that was collected from the University of Miami campus. We had a very aggressive testing, tracking, and tracing program on campus. So we had very in-depth data on the prevalence of disease on campus, and we could use that to look at the wastewater and compared it to the wastewater in individual buildings and on the various campuses, the Gables, the medical, and the Marine campus. We also had access to university hospital data through their electronic medical records, we were able to get information on comorbidities. At the county level, the Florida Department of Health had positive cases on a zip code basis. And then at Miami-Dade County Public Schools, through our RADx UP supplemental funding, we were able to get information on absenteeism. Amongst all of these different sources of information, I think the most critical and the most extreme when it came to lockdowns was the information from the hospitals. The lockdowns were initiated because the hospitals were overwhelmed. And what we find is that wastewater-based methods of analysis can predict hospitalizations into the future. Here we have a black line showing the hospitalizations over time in Miami-Dade County. And then we have the levels at the regional wastewater treatment plant shown in the green circles. And as we can see that both of them tend to follow one another, with the wastewater slightly ahead of the hospitalization numbers. What we see early during the pandemic is that low levels of SARS-CoV-2 in the wastewater resulted in large number of hospitalizations. But as the pandemic progressed, as we had vaccines, the numbers decreased in both the wastewater and also in the hospitalizations. But then as the virus evolved, we had increased transmission. So then we're seeing other waves come through, such as the Delta wave and the Omicron wave. So as the pandemic progressed, early on, the wastewater, small levels in the wastewater resulted in high levels of hospitalizations, but then the slope tended to flatten as we went from the Delta wave, Omicron, to the post-Omicron wave. And right now where we are is we have high levels in the wastewater, but low hospitalizations. In addition, looking at the variant data was very interesting. On the right panel, we have the top colored panel is clinical data. And then on the bottom, we have the wastewater data. The different colors represent the different variants as we observed in both the clinical and in the wastewater sample. And if you look at the color change in both media, you can see that they both followed each other pretty well. In addition to that, what made the wastewater so fascinating is that sometimes you are able to observe the variant in the wastewater before we observed it in the clinical samples. So for example, during the Delta wave, we observed the Delta variant in the wastewater a week prior to having it observed in the clinical samples within Miami-Dade County. So it was a pre-warning not only of SARS-CoV-2, but also of the variants that were coming through. In terms of the hospital wastewater, the levels of SARS-CoV-2 in the hospital wastewater, we got variable results on that. In terms of the hospital wastewater, of course, it did correlate with the number of positive patients within the hospital. And early during the pandemic, we did see correlations with remdesivir administration within the hospital. But beyond SARS-CoV-2, we also looked at additional targets such as Mpox. In the university hospital, we have records of the number of clinical cases within the hospital during the June through October 2022 timeframe. We also have wastewater samples from the hospital, and we were able to see a correlation or a coincidence in time. We saw the elevated levels in the hospital, also seeing the Mpox within the wastewater at the same time. Also at the university hospital, there was a lot of concern, at least in Miami, about Candida auris. Candida auris is one of those infections. People may come into the hospital, they may get it while they're in the hospital. And because of the difficulty in treating it, people remain in the hospital for long periods of time. And at the University of Miami, there was a lot of interest in Candida auris because we had patients that were in the hospital for up to a year at a time because of this infection. And you can see within the university hospital, we had Candida patients shown by the blue, the number of patients shown by the blue line here. The orange symbols are the PCR detections from the wastewater. Mark Sharkey from the Center for AIDS Research developed the PCR technique for detecting Candida auris in the wastewater. But what's also very interesting is that not only did we look at it molecularly, we could detect it molecularly, but we could also culture it from the wastewater, as well as you can see in the chrome agar plates shown to the right. So our next steps are now focusing on looking at additional targets. In addition to University of Miami and Weill Cornell Medicine, we have an additional partner, Yale University, that is working with us on our samples. Then there are also Wastewater Scan and BioBot, which have national programs to look at pathogens in wastewater. So we have 4 laboratories that are looking at the wastewater, that are sharing wastewater essentially. But in addition to looking at SARS-CoV-2, amongst the different laboratories, also looking at influenza, AB, poliovirus, RSV, norovirus, Mpox, human metapneumovirus, and Candida auris. But to look at the comparison between the laboratories, amongst the 4 laboratories, here we have the positive cases shown by the bar plots with the number of positive cases to the left-hand side over time within Miami-Dade County. And what we know is that over time, as the pandemic progressed towards the end, there were fewer and fewer cases being reported, but it's mainly because people were not being tested or not reporting the results of tests. But if you convert the data from cases to positivity, divide by the number of tests, take the cases and divide by the number of tests, we still see that the positivity, those who do get tested, the positivity still remained high and was coming in waves through the wastewater. So if we look at the wastewater and the various laboratories, we can see that the 4 different laboratories were pretty much following the positivity as observed here. It was being followed through the Delta wave, followed in the Omicron wave, and the various post-Omicron waves. However, if you're going to compare one laboratory to another, the absolute numbers do not match up, but the overall trends do seem to follow one another. So as long as that one laboratory you're using the data from that same laboratory, the trends over time are pretty matched up, well matched up. So in this project, we can see the matching of the data in terms of time series. Also from the shotgun sequencing. This is data that came from Weill Cornell Medicine from the shotgun sequencing results. And what was fascinating is that from the shotgun sequencing, we have pathogenic viruses that could be observed as shown on the left, and we have the pathogenic bacteria that are shown on the right. We're able to very interestingly be able not only to see the communities, the microbial communities, but now we can get down to the level of specific pathogens. So what was fascinating about the metatranscriptomic analysis is that now we can even see pathogenic viruses such as norovirus and Aichi virus, and the pathogenic bacteria are shown on the right-hand side as examples of the depth to which the data is now available in terms of coming up with an agnostic method to look at the wastewater samples. In addition to pathogens, we're also able to see antimicrobial resistance genes from the wastewater. What was interesting and has been known in the literature is that hospital wastewater tends to have higher numbers and diversity in antimicrobial resistance genes. But what we found was very interesting is that as we went down through the sewer system from the individual buildings, which we call dormitories, all the way to the campus basin, to larger and larger basins, all the way to the wastewater treatment plant, we're losing the number of antimicrobial genes, and we're losing some of that diversity. It's common wisdom to know that wastewater treatment plants are a source of antimicrobial resistance, but we were just not aware of the amount of AMR in the upstream parts of the wastewater as well. We also conducted host attribution in collaboration with Phase Genomics. And you cannot see the lettering here, but on the vertical axis, we have different genus of bacteria. On the horizontal axis, we have the AMR genes. The yellow corresponds to AMR that were associated with plasmids, and the blue corresponds to AMR that was integrated into the genome. So for example, for the genus of Provotella, there were 2 blue lines. So there were 2 AMR genes that were integrated into the genome. And we feel that this kind of information is very valuable to the clinical community, knowing which bacteria and what AMR genes are being carried within the community, within the wastewater, can be used to help in determining the best type of antibiotics to prescribe to the population if we have better information from the wastewater. So possible applications to consumer products. We're looking at liquids, wastewater is a liquid. And again, we know that the technology now has advanced to the point that we now have these agnostic methods, as mentioned in the previous presentation. We can use it to look at, for example, these are just some ideas, the wash from produce. So not only can we do PCR, but we can potentially look at sequencing to get an agnostic method on the wash waters. As mentioned by Commissioner Califf at the very beginning of this conference, there was concerns about H1N1. And for example, the same technology can be used to look at runoff, to identify sources upstream, to see if the animals that are contributing towards that runoff could potentially be carrying H1N1. We also know that the sensitivity for pathogen detection is now increasing and much better than it was in the past. And these increased improvements in the technologies can be also applied to food product screening. We also, within the shotgun sequencing, very interestingly, we were able to look at we were able to get information about animal RNA. And we believe that that was due to the diet of the people that were contributing to the wastewater. So we can use the wastewater to look at community diet, and the same technology can be also used to look at food authenticity. And then, of course, these technologies have been used significantly in the past to look at sources of outbreaks, and this, what we've learned through wastewater, just further emphasizes the ability and the power of this technology to identify strains and can be used in sourcing the outbreaks of sourcing outbreaks. So our next steps are shown here, and the next steps are to focus in on targets beyond SARS-CoV-2, additional pathogens, and also expansion of the workflows to not only include wastewater, but air and surface sampling. We have done some air and surface sampling and believe and have shown that actually the air and surface sampling is also capable of expressing what may be going on in the community in terms of disease transmission. We see incredible potential for target agnostic approaches, such as the sequencing, as mentioned before. We can see the whole community without knowing what may be there. We can compare the target agnostic results to baseline data, which will help us to identify which organism may be a concern at any given time. The big challenge with all of this is to define what it all means. We can do a lot of measurements, data, but it's important that it all be compared against clinical data so we have a meaning to the numbers that we see in the environment. We also believe that there's important steps in automating. We have so much information, but it needs to be assimilated quickly. The bioinformatics, if they can be done automatically and generate reports would be in real time, would be a very important aspect to implementation of this technology during outbreak periods. And then we also believe that this technology that we learned from wastewater, we emphasize through wastewater, can also be expanded for consumer products. So at this point, I'd like to thank the audience, and also our references are listed here with the DOIs in case you want to get into more detail. So thank you again. And at this point, I'll turn it back over to the moderator. Thank you.
Thank you, Dr. Solo Gabriel. It is now my pleasure to introduce our next speaker, Dr. Dan Cornforth of the Centers for Disease Control and Prevention. Dan, please go ahead and start sharing your slides now as I briefly introduce you. Dan Cornforth has a background in public health, mathematical modeling, and microbial genomic analysis, and currently serves as the lead for the Data Analytics and Visualization Unit in CDC's National Wastewater Surveillance System, or NWSS. Dan holds a Bachelor of Science in Mathematics from the University of Texas at Austin, as well as a Master of Science from the University of Oxford and PhD from the University of Edinburgh, both in biology, where he focused on the mathematical modeling of the transmission and evolution of bacterial pathogens. He then completed a postdoctoral fellowship in microbiology at the University of Texas at Austin before becoming a research scientist at Georgia Tech, where his research centered on bacterial gene expression in human infections. Dan joined CDC as an Epidemic Intelligence Service Officer, supporting the National Syndromic Surveillance Program, and later transitioned to his current role in wastewater analysis and NWSS. Today, Dr. Cornforth will explore how wastewater genomics can enhance public health surveillance, offering early warnings for emerging variants of concern. The CDC's NWSS collaborates with over 30 laboratories nationwide to sequence SARS-CoV-2 and wastewater samples, providing insights into circulating variants. Dr. Cornforth, I'll hand it over to you, and thank you for joining us today.
Very much. Are you able to see my screen?
Yes.
All right. So, yeah, I'm going to be talking today. I just realized. Hey, yeah, I'm going to be talking today a little bit about genomics at the National Wastewater Surveillance System, or NWS. So, for a lot of infectious diseases, the affected individuals can be thought of like an iceberg. So on the bottom, there's all the infected persons. Some of those individuals will end up getting clinical tests. Some of those will end up as cases. Some of those will end up more severe as hospitalizations and intensive care and even deaths. There's different surveillance systems that are meant to address these different steps along this iceberg. But one thing that's often difficult in public health is understanding what happens before individuals seek clinical care, when individuals might be asymptomatic, when individuals might not have access to care. And this is a unique role for wastewater, or at least a rare role for wastewater, because it can complement other systems. It offers a more complete picture of infections along with the other data sources, except there's a lot of untapped potential, particularly on the genomic side of this. So I'm going to talk through a few advantages of wastewater surveillance. First of all, it's efficient. It's an efficient method for routine surveillance, and it's economical. So rather than having to individually sample and sequence clinical specimens, having it pooled allows for a lot more economical understanding of the infection situation in a given community. As I mentioned before, it also captures asymptomatic and pre-symptomatic individuals, as well as mild infections. This matters because some individuals aren't going to engage in health-seeking behavior, and they might not have test access. And beyond this, some individuals will, at the beginning of SARS-CoV-2, the pandemic, individuals were going to the hospital to get clinical tests to determine whether or not they actually had SARS-CoV-2. But then later on in the pandemic, this shifted toward more at-home tests. This didn't really affect wastewater in the way that it affected a lot of our other surveillance methods. And lastly, data is very timely, so it can be available within days of shedding onset. So with that, there's a few important limitations of wastewater surveillance that are worth mentioning. One are geographic limitations. So 20 to 25% of of the country doesn't feed into sort of centralized wastewater systems. So these are mostly septic tanks. There's also the fact that sewer sheds, the boundaries don't necessarily align with other geographic boundaries. So for example, the area circumscribing that is served by a particular wastewater treatment plant might differ from zip code boundaries or from county boundaries, which can make analysis more challenging. There's also a lot of variation among wastewater systems. This can be first in part from population sizes. So at a given area, at a given treatment plant, you can have the population served varying pretty dramatically just by fluxes of populations. For instance, students that might come to a university and then leave for the summer, or tourism, for example. Also, the inputs can vary considerably in terms of how much rainfall there might be or other inputs into the system. And it's important to note that there can be misinterpretations, and there sometimes are. So one thing that's worth mentioning is that negative results don't necessarily indicate the absence of infections, and also that you can have animal waste in other products. And this has become, a common talking point for, some recent data for H5, for instance, trying to distinguish what might be an animal contribution from what might be a person contribution. So this slide just shows the overall expansion of news from when it started in 2020 toward the beginning of the pandemic to the current day. And so you'll note and you'll see pretty clearly that we've expanded a lot. We're currently at about 1,500 sites in 50 states, 6 territories, and 6 tribal communities. And this includes 250,000 unique wastewater samples and over 100,000 sequence samples. And this represents about 40% of the population. And there's a growing list of targets. So currently, that includes SARS-CoV-2, Mpox, flu, RSV, and others. But like I said, this list is expanding. So there's a lot happening at the national level or at news in terms of the targets that we're able to address. So this shows an overall view of wastewater information starting at the community level. So it's all community based. Obviously, individuals create waste. And then there's sampling. So this sampling occurs at wastewater treatment plants. And then it's processed. So this can occur and tested. So this can occur at laboratories, health departments, private, commercial and academic entities. And this is where, the testing actually happens before it's sent on. Before it's sent on to STLT health departments, and then to CDC, and then our academic collaborators, and also our centers of excellence. And I'll just say really quickly, our centers of excellence, this is a system that we have 6 of these centers throughout the country that help us sort of push forward and innovate wastewater. And so we rely on them a lot for input and to really push the boundary on some of what we're doing. So this is just an overview of the news laboratory capacity. The first point is CDC wastewater laboratory. So this is what's actually happening in CDC. It includes test development and validation and assay development. Some of this occurs at CDC and some of this is contract-based development. Perhaps more important is the news laboratory network. So this is really what we rely on for all of our samples, a lot of the innovation, like I said, and really it's the core of what makes news news. So this consists of 114 labs overall, most of which are public health labs, and 31 of these have sequencing capacity. The data is then submitted through a variety of mechanisms. It can be manual or API or SFTP into a system that we call Decipher. So Decipher is a Palantir platform that we use for a lot of our data analysis, basically every step of the data pipeline, from ingestion to analysis and also visualization. It's a central component of what we do. It's cloud-based and allows for the automation of a lot of our processing because, as you can imagine, we get a lot of data coming in. Just on the sequencing side alone, obviously, those files can be quite big. And so having this system is really helpful. I'll say here that the 2 main types of data that we ingest can be both concentration and sequencing data. So I'll focus mostly in the latter part of this talk on the genomic side, but I'm going to talk a little bit about concentration as well. Something that's worth mentioning is that the results for these can be linked. And so the same sample that we have concentration information for, we also have sequencing information for. So, and to be clear, there's some concentration samples, most even, that don't have sequencing associated with it, but we're able to pair those, and that can allow for some interesting comparisons, some useful comparisons for public health. In addition to that, this system, all the data that we get for sequencing, we're able to put onto a bioproject on NCBI, and I'm going to talk a little bit more about that. So basically all the data that we get, we're trying to be as transparent and public as we can about it. So this is the news Decipher dashboard, and this is sort of a core of our communication and sharing of information with our jurisdiction partners. So this just shows a kind of central dashboard that includes information on percentage change, percentiles of a given virus, proportion of the text, as well as variant-specific metrics. So this map has various layers of it, but and you can toggle between them for different targets and for different particular metrics that you're interested in. So we use this again within CDC and then all of our jurisdiction partners. And here's the same thing, but just showing some of the sequence visualizations in particular. So on the left side, you can see a map with these different points on it. These different points for SARS-CoV-2 variants, and they correspond to these different colors here. So on the right, this is just another visualization that we have available. And this is for our jurisdiction partners to be able to see how their different data types. So the concentration data, which is, this curve, as well as the cases laid on top of the different variants, which is on the bottom. So the point is that it allows an easy integration of these different data sources that that folks can see their data and can see kind of how all these parts are fitting together. So I mentioned before that we have a big emphasis on our public-facing data. So right now on data.cdc.gov, you can get a full history of our COVID data. And this is an expanding list. So we hope in the next several weeks to expand the targets that we're now showing. So for instance, we are in the process of sharing publicly additional information on flu and RSV. So those will likely be short follows on what we're going to be able to display. In addition to that, all of our sequencing data is put into this NCBI bio project. And again, this is over 100,000 files in here. So this just shows kind of a more granular view of our data flow from when we get them from either utility partners, departments of health, academic partners, federal partners, commercial partners. So for the sequencing data, this can be sent to news via Decipher. It can also be sent directly to NCBI. And regardless of which path it goes, it's going to end up on both at the end of the day. So all the data that we get, we put immediately onto NCBI, and anything that is put onto NCBI, we pull. And then we have it on our platform for this integrated analysis. And this accepts various sequencing platforms as well as different tiled amplicon schemes. So this is an overview of our analysis pipeline. And one reason that I wanted to put this in was to sort of give a shout out to FDA, because this work was based closely on a previous pipeline of theirs called CWAP. And we basically took that and we streamlined it a little bit. We maintained the tools that we needed and just kind of made some tweaks basically, and then put this on our Decipher platform in a single Docker container. So this allows us to update and not have to worry about some of the issues that we were running into previously. At the heart of it is Freya for variance abundance estimation. And I'm not going to go too much into the details here other than to say it's accepting different read inputs that can be as a raw FASTQ or in certain cases a BAM file. Then there's an alignment step and then just some additional processing before it gets to Freya, which is the core of the output that we're doing. And here's just some examples of output. So on the left, there is the dominant variant of concern, and this is just for different sites. And so you can see over time, those dominant variants of concern shift for the different sites. On the right, this is just showing a jurisdiction. And this is showing just a stacked bar chart of those variants over time. And as I mentioned before, we do have a big emphasis on public-facing wastewater data. And this is something that we're continually trying to share more and more as we're able to. So this first one is COVID Data Tracker. This has been around for some time, and it'll show a map with all of the variant information on it. And then in addition to that, this is the variants, just the COVID variants that's now appearing on our website. And I'm going to just dive into that a little bit more closely. So this just shows these figures that I'm sure y'all have seen a million times, but just shows at the national level, these variants. And so it's just aggregated up right now. So these are just available on our website. And these are figures, and some of these figures, some of these analyses are things that we're constantly working on improving. And so we do hope to have more to share in the coming months. So I'm just gonna give an example of a success story for wastewater genomics. This is an early one, but it's representative of several that followed. So this is for Omicron, and some of the early detections were in wastewater. So Omicron associated mutations were seen before there were clinical cases identified. And, this detection allows for variant tracking and can complement the clinical testing. So it can allow for early detection of emerging variants. And as I mentioned, this is just one example, but we've seen this happen in BA 286. We've seen this happen in JN1 more recently. And so it often is becoming an early signal for, potential variants. And this is something we'd like to lean into a lot more, but this is, it's already showing, pretty strong indicator that it's going to be able to find things relatively fast and can really complement other work going on. So I'm going to close just talking a little bit about some next steps for us, sort of what's on our radar. So this first one is, more work in methods and streamlining workflows. So a lot of this work is going on at our COEs and our centers of excellence. And some of this is developing and trying different sort of sequencing approaches. Some of this is, communicating, trying to figure out what works best for which pathogens, which ones might be easier, which ones might be harder, in different approaches. So, tiled amplicon sequence genome-wide, or, specific targeted amplicon sequencing, and trying to understand kind of what is of highest value. Secondly, trying to reduce cost and time. So this is something that we've been working on a lot, is trying to improve the turnaround time for our sampling so that we can get that information as soon as possible. And as I mentioned, trying to figure out what the best way of processing so that jurisdictions can afford it and that it makes sense in everyone's budget and their plan, that it's actually showing the results that they need in order to keep doing it. So next, the increases of targets. So this is with every outbreak response. Once we can produce concentration data, the next question we have is it possible to sequence? And so there are a lot of conversations going on to try to understand that a little bit better about, SARS-CoV-2 is what we have up now. But as some of y'all know, there's some things that make SARS-CoV-2 a little easier than some other pathogen targets. And so trying to understand that, and also trying to develop species agnostic approaches as well. And expand coverage. So this is something that we are constantly trying to do, because obviously the more coverage we have, the more we're able to detect emerging threats. And so we have a national contract that can onboard up to 400 sites to help fill in gaps. And then we're constantly working with jurisdictions to improve our coverage as well. As I mentioned before, continue and improve some of our open data sharing processes. This is sort of core to any federal agency, is to make sure that we're able to give that as an asset to academic researchers as well as the public. And lastly, communicating effectively the value to the public, making sure that they understand that how to use wastewater, how to interpret it, so that it's not just some weird thing that some people read an article on from time to time, that it's actually core to informing decisions from the average person. So I think that that's all. Thank you so much for listening. And now I will return.
Thank you, Dr. Cornforth. As our final speaker for today, I'd like to introduce Lauren Ahart, also from the Centers for Disease Control and Prevention. Laura, please go ahead and start sharing your slides while I do a brief introduction. Lauren Ahart is a surveillance epidemiologist in the Parasitic Disease Branch, or PDB, at the CDC. Lauren began her CDC career in 2017 as an ORISE Fellow on CDC's Division of Foodborne, Waterborne, and Environmental Diseases, where her projects and work were focused on antimicrobial resistance in foodborne pathogens. In 2020, she joined PDB to work on cyclosporiasis surveillance, outbreak investigations, and response activities for PDB. In addition to her work on cyclosporiasis, she also assists with surveillance and technical support for epidemiological investigations of trichinellosis. She will present the epidemiology and surveillance of cyclosporiasis in the U.S. and describe the current genotyping tool. This presentation will highlight recent outbreak investigations and demonstrate how genotypic data have been used to inform epidemiologic cluster and FDA traceback investigations. Lauren, I'll hand it off to you, and thank you for joining us today.
Thank you so much, Dr. Mercado. Could you confirm that you can see the correct slides, the correct view?
Yes, we see them.
Perfect. Thank you so much. So good afternoon again, everyone. My name is Lauren
I'm
Ahart.
sorry, Lauren, we're seeing the, we're not seeing the presentation view.
Okay.
Yeah, we're seeing your
No
notes.
worries. I will attempt to swap this.
There
How
we
about
go.
now?
we see
Okay.
them.
Sorry about that.
Thank you.
So my name is Lauren Ahart, and I'm an epidemiologist in CDC's Division of Parasitic Diseases and Malaria. Today, I will be presenting an overview of cyclosporiasis surveillance and genotyping in the U.S. So I'll start by providing a brief background on cyclosporiasis, and then I will share more about our U.S. cyclosporiasis surveillance genotyping and how we apply genotyping in the U.S. during cyclosporiasis outbreak investigations. So to give everyone a little bit of background on cyclosporiasis, it is a gastrointestinal illness caused by infection with the parasite Cyclospora cayetanus. For U.S. infections where we've been able to identify a source, it has historically been associated with international travel to areas of endemicity or consumption of fresh produce imported from endemic areas. The average time for symptom onset is about a week, but can range anywhere from 2 to 14 days. Also, the most common symptom is watery diarrhea, but there are some other noteworthy symptoms such as loss of appetite, weight loss, cramping, bloating, increased gas, nausea, or fatigue. The first cyclosporiasis outbreak was documented in 1990, and outbreaks have continued to occur almost every year. So this is a brief overview of the life cycle of Cyclospora. The Cyclospora cayetanus life cycle begins and ends with humans. So the first phase begins when an infected person excretes unsporulated oocysts, which are non-infective, in their feces. And so those oocysts then sporulate or mature in the environment. And depending on the environmental conditions, it can take about a week or two for oocysts to become infective for another human. So the sporulated oocysts then enter the food chain, and people become infected with Cyclospora by consuming contaminated food or water. Therefore, direct person-to-person transmission is highly unlikely. Unlike bacteria, Cyclospora reproduces sexually, which is an implication for genotyping. You'll hear more about that later in the presentation. But just to provide a quick example, 2 different case patients infected with Cyclospora may have a common source, but could appear to be unrelated, and multiple genotypes have been detected. And then finally, Cyclospora is diagnosed by identification of the parasite in stool specimen microscopically or by detection of DNA by PCR. So now I'll talk a little bit more about cyclosporiasis surveillance here in the U.S. Cyclosporiasis has been nationally notifiable since 1999, and it is reportable in 46 states, Washington, D.C., and New York City. Cases of cyclosporiasis are reported to local and state health departments after a sick patient visits the doctor and is diagnosed by a physician. So our surveillance for cyclosporiasis is a passive surveillance system because we rely on patients to go to the doctor before they can be diagnosed. It's also important to note for U.S. cyclosporiasis surveillance that cases are reported by state of residence, which may not be the same places where their infection was acquired. So we conduct surveillance for cyclosporiasis year-round, meaning state and local health departments can interview and report new cases to CDC at any time. The Cyclosporiasis National Hypothesis Generating Questionnaire, or CNHGQ, is the primary data collection tool used to gather information on case patients. So the CNHGQ is a hypothesis generating questionnaire that collects information on travel history, events, and reported food exposures during the 2 weeks preceding illness onset. These extended EpiCase data are then used to identify common exposures among cases. There has been a seasonality seen in areas where cyclo has been studied, and we also see this in the US as well, where 90 to 95% of cases each year occur during the summer months. So we, along with state health departments, conduct enhanced surveillance during the cyclosporiasis peak period, which is May through August. Some of our enhanced surveillance activities include actively following up with jurisdictions on domestically acquired cases, collaborating with jurisdictions as well as FDA to investigate epidemiologically defined clusters and outbreaks, and finally conducting genotyping activities. So, as I mentioned, our cyclosporiasis surveillance in the U.S. focuses on domestically acquired cases, which are laboratory confirmed cyclosporiasis cases who do not report travel outside of the U.S. or Canada during the 2 weeks before their illness onset. We focus on domestically acquired cases primarily because those cases are more likely to have consumed fresh produce that was either imported or grown here in the U.S. Therefore, as public health authorities, can intervene and potentially take action to prevent further illnesses from occurring. So now I'd like to provide a brief overview of the CDC cyclospora genotyping tool in our methodologies. So I'll first start by saying the whole genome sequencing, or WGS, is not feasible for cyclospora, which is why we developed the CDC cyclospora genotyping tool. The CDC cyclospora genotyping tool can be used to identify cases that are genetically related in outbreaks using the combined lab and epi data. So this slide provides an overview of our CDC laboratory methods, which consist of deep amplicon sequencing of 8 genetic markers. To briefly explain this process, the state public health labs sends positive stool samples to CDC to be genotyped, and our CDC lab team receives and processes the specimen in-house and sequences them using 8 genetic markers before feeding it into a bioinformatics algorithm to identify genetic linkages. Their goal is to also determine the probability that cases are genetically related. And so there's no direct comparison with matching of genotypes done in this process, which means there's a little bit more uncertainty compared to bacteria, where comparison is done by whole genome sequencing. So our primary goal in using the genotyping data is to identify individuals who were likely sick during the same time frame. We use specimen collection date as a proxy for illness onset in our genetic clustering analysis, in addition to a temporal component or a 2-week sliding window, which I'll discuss in more detail here in a bit. Specimen belonging to the same genetic cluster can also be assigned what we call a temporal genetic cluster, or TGC. And in order to be assigned to a TGC, first, again, specimen must belong to the same genetic cluster. There must also be at least 3 specimens total to form a new TGC, and they must also be from at least 2 or more jurisdictions because we're really interested in those multi-state outbreaks. And finally, those 2 or more isolates from 2 or more jurisdictions must have collection dates within 2 weeks of each other. Once those criteria are all met, the specimen is assigned to a TGC, and then the TGC is defined where we look for additional specimen within the genetic cluster that have collection dates within a 7-day sliding window. And we do this process iteratively. So now I'll share a little bit more about our genotyping data and how they are used and can be useful for outbreak investigations. So I'll first point out that cyclospora genotyping are not currently used for cluster detection, and outbreaks are still being defined epidemiologically. Clusters and outbreaks are primarily driven by epidemiologic data, and we still need those data to identify a suspect vehicle during an investigation. Nevertheless, the genetic data can be really helpful in outbreak investigations. Genetic data can provide objective evidence for linking cases in epidemiologic clusters, and it can also confirm whether epidemiologic clusters are related or not. So now I'll briefly touch on CDC's epi data analysis methods. After the CDC lab team completes their genetic data clustering analysis, our epi team reviews the epi data and examines the temporal genetic relationships. The purpose of the epi analysis is for us to identify commonalities in the food exposure, stores, and restaurants reported. However, there are a few other steps that I'll briefly explain now with you. So if you look at the diagram on the far left side of the screen, it represents the temporal analysis performed by the CDC lab team. Here, the lab team assigns what we call a TGC code to each newly identified temporal genetic cluster. So for example, the first TGC code identified this season was 2024-001, and then subsequent clusters were numbered sequentially. The CDC lab team then shares a list of all of the successfully genotyped specimen with us on the EPI team. And then we look at the case IDs and analyze those data in further review. And then once we kind of have a chance to analyze those data, we summarize them and share them back with our state partners. So in the next few slides, I wanted to walk through a few examples of how the cyclospora genotyping data can be helpful for epidemiologic cluster investigations. The first example that I'll share is a leafy green cluster identified by multiple states in 2022. So in June 2022, a cyclosporiasis cluster was identified in a correctional facility, and the suspect vehicle was determined to be a salad mix produced by producer A. The ingredients of the salad mix made by producer A included iceberg lettuce, red cabbage, carrots, and carrots. Sorry about that. So in July 2022, a cyclosporiasis cluster was identified in a hospital in a different state, and the suspect vehicle was determined to be romaine. Also in July 2022, a cyclosporiasis cluster was identified in a third state and linked to Caesar salad kits also made by producer A. So the suspect vehicle in this third cluster was also determined to be romaine. And additional clusters were identified in other hospitals and long-term care facilities, but we ultimately weren't able to identify a suspect vehicle due to poor patient recall for those clusters. Nonetheless, at that point, we had 2 clusters where the suspect vehicle was said to be romaine. And given this information, FDA initiated traceback investigations on romaine in July 2022. Later that month and into early August, the genotyping results became available and proved to be informative. So we saw in the genotyping data that cases linked to the correctional facility where the suspect vehicle was a salad mix made by producer A actually genotyped into the same TGC as cases linked to the Caesar salad cluster where the suspect vehicle was romaine made by producer A. And for reference, in case you're interested, the TGC code is here in orange on this slide. So it's TGC 2022-001. On the other hand, cases linked to the healthcare facility, one of the healthcare facility clusters was identified romaine as the suspect vehicle, and they all typed into a different TGC. And so that's TGC 2022-012. So as you can see here, the two epidemiologically identified clusters, the genotyped into 001 were both made by producer A, and the suspect vehicle was romaine, whereas the cases that typed into 012 typed separately. And as I mentioned earlier, traceback investigations were initially initiated on romaine. However, the genetic data showed that the romaine clusters were not likely related. And so because of this, the traceback investigations were actually reorganized to combine the Caesar and the correctional facility clusters based on that common producer A, which was separate from the healthcare facility cluster. So now I would like to share how the genetic data can be used to highlight that seemingly unrelated epi clusters are likely related. So this example that I'll share next is from 2023 and includes 2 states with restaurant-associated clusters. So in June 2023, a large cluster of illnesses was identified and linked to a single Mexican-style restaurant. Despite extensive patient interviews, no suspect vehicle was identified at that time. Also in June 2023, a smaller cluster of illnesses linked to 2 American-style restaurants was identified in a different state. So the 2 American-style restaurants had a common owner and shared staff and suppliers. However, a suspect vehicle could not be identified among cases linked to that cluster. Ultimately, there was no indication that these 2 clusters appeared to be related until the genotyping results became available in July 2023. So ultimately, the specimens submitted for cases linked to the Mexican-style cluster genotyped together into TGC 2023-011, and the American-style restaurant clusters all genotyped together into the same TGC as the other restaurant clusters. So again, into TGC 2023-011. So the 2 states associated with these clusters compare ingredient lists and suppliers for the restaurants. And leafy green A was the only ingredient in common, and the restaurants in both states had the same supplier, which was epidemiologically plausible. So in summary, overall, our genotyping data can be informative for cluster and outbreak investigations, as it can provide evidence that cases that genotype together likely share a common source of infection. It can also help prioritize cluster investigation activities such as case follow-up. Epidemiologic data still remain very valuable for identifying a suspect vehicle. However, there can be some challenges in collecting those data as well. For example, case follow-up to request additional information on reported food exposures is not always feasible. And additionally, patient recall on fresh produce consumed during the 2 weeks before illness onset can also be challenging. As we move forward, we continue to take our combined genotype and epi approach to yield the most impactful public health response to cyclosporiasis outbreaks. And this work would not be possible without the help of our partners at the state health labs and also the public health labs, as well as our partners and colleagues at FDA. And then I'll finally say a huge kudos and thanks to our internal teams, both on the EPI and labs side here at CDC. And thank you to you all for your attention this afternoon and the opportunity to share our work. And that's all I have, so I'll pass it back over to Dr. Mercado.
Thank you, Lauren, and thank you to all our speakers for their excellent presentations. With our remaining time, we'll move on to the Q&A portion of our session. We do have time for a few questions. I will ask Dr. Jackson to please join me here at the front for the Q&As. And for our audience in the room, please step up to the microphone to ask your question. For our online attendees, please use the Q&A box in Zoom. And prior to asking your question, we do ask that you please state your name and affiliation. Thank you. So I don't see any questioners come up to the front. I do see in our Q&A box there was a question answered live by Dr. Solo Gabriel, I believe. So Dr. Solo Gabriel, if you could please reiterate the question and the response that you gave.
I'm looking, I lost the question when I clicked live, but the question had to do with whether or not H5N1 can be detected in wastewater. Although we have not, at Miami, have not been specifically looking for it. Wastewater, I was at a presentation of Wastewater Scan, where they have been detecting H5 in wastewater from municipal wastewater treatment plants. And they attribute that to possible human source or even infected animal products. What I was proposing was to look at runoff from agricultural locations to potentially get it directly from the feces of the animals, as an example. So yes, I think it is possible, and there is evidence that it can be done.
Great, thank you, Dr. Solo Gabriel. I don't see any other questions in the chat, so I think I'll go ahead and ask one question directed to Lauren. Have you observed the same Cyclospora genotypes appearing each year? And if so, are the genotypes associated with specific fresh produce items?
Yes, thank you so much, Dr. Mercado. Yes, I can say that we have seen some genotypes that appear, year to year. However, we have not yet looked into whether or not those genotypes are associated with specific fresh produce items. And that is something that we're interested in doing and hope to do in the near future. We just have not had the opportunity to really dig into those data just yet. But yes, we do see some genotypes from year to year.
Thank you. I have a question for Dr. Jackson. If you could speak more about the challenges surrounding standardization of these detection technologies that are being brought out to the field, so particularly in situations where rapid detection of the biothreats is required to inform decision-making.
I think NGS, untargeted NGS for biosurveillance has amazing potential because, again, you already know what you're looking for. Typically, you know the genotype of what you're looking for. You're looking for these genetic signatures, and it's an amazing technology for doing that. One of the challenges is validating every possible organism that you want to detect, which there could be hundreds of different species of viruses and bacteria, to validate them in a rigorous way. And that's really what's been holding up the regulatory approval processes for using NGS as an infectious disease diagnostic, because it's not clear exactly what a validation approach might look like when you say that my technology can detect anything. Normally, if you have a targeted technology that detects salmonella in stool, you validate against salmonella in stool. But if you say I can detect anything in stool, that raises some validation questions that the industry and the regulators and NIST are still working to figure out.
Yeah? Okay.
Thank you. If we have time for one more question, I'll direct one to Dr. Cornforth. So, Dr. Cornforth, can you speak more about the socioeconomic implications surrounding wastewater detection? So, Dr. Chatterjee kind of touched on this in the keynote this morning, but this would be regard to misuse of human genetic information that would be gleaned from wastewater surveillance, as well as stigmatization of whole communities wherein surveillance data points to specific pathogens that are spread in those communities.
Yeah, so with respect to the stigmatization, this is something that's always on our radar. And so when we're reporting our data, this is always, we're very careful about the size of the contributions toward the sewer shed that we report. So we won't report anything less than a few thousand, for instance, to make sure that, there aren't as big privacy concerns about the stigmatization. In terms of the privacy issues in the data. So, I mean, right now we use, it's just held amplicon on SARS-CoV-2. I think that is going, and there might be mispriming, so you could get other reads in there. We do a process of scrubbing, and that scrubbing for any human reads happens before we receive that data also, and then we do it again. So, we have processes to prevent any of that sort of high privacy information from being in those samples.
Thank you very much. Okay, so I want to say thank you again to everyone in this session. Thank you for questions and discussions. This concludes our last session, and we'll wrap up the day with closing remarks from Dr. Alexis Norris, who is the co-chair of the FDA Omics Working Group. Thank you.
Hey everyone, I'm Alexis Norris. I'm a bioinformatics reviewer over at the Center for Veterinary Medicine and also co-chair of the FDA Omics Working Group with my wonderful colleague, Matt Hartog, who has been working hard today at the computer, making sure we don't have any technical glitches. So this year we decided, this is our second year, and we decided to add a poster award to the session, kind of in the spirit of what we have seen at Scientific Computing Days. And so we're going to have two awards. The first award is the Peer's Choice Award. And so thank you to everyone who attended in person. Those of you online, hopefully we can see you next year and come in person. And so the winner of this award was selected by Omics Days attendees. And this award is presented to Kentaro Toma and Arya Eskandarian for their poster titled Visualizing the Evolution of RNA Viruses, a web-based platform for tracking genetic and antigenic changes. If Kentaro and Arya are in the room, we have an official little award. If not, we will find a way to get it to you. And then if the co-chairs, the event co-chairs want to come up for awarding the next award, Lerna, Rick, and Paula. Yep. So the second award is the Omics Days Co-Chairs Award choice. So these three wonderful people you see have helped put together this event, and so grateful for all three of them. And the winner of this award is awarded to Mayumi Miller for her poster titled Large Alterations in the Genomes of Pigs Edited Using CRISPR-Cas9. So if Mayumi is in the room, if you want to come up and get your beautiful official little award. Thank you to our photographer, Hermes. And so that concludes our event. On behalf of all my organizers, all the session moderators, co-chairs, I'd really like to also thank our presenters for their talks, for taking the time to come present and discuss. And to our audience, thank you. Thank you for holding out till the end. If you are one of our FDA colleagues, we're going to be continuing the event tomorrow over in Building 2, room 2047. And if you're not an FDA colleague, we'll see you a year from now. Really look forward to that. And we've covered the main agenda, and We encourage you to connect with each other. We'll see you tomorrow. We'll see you next year. Thank you.